used in treating dementia. So compare it for the comparison studies, as it goes hand in hand. That's the reason I have selected both the drug which comes under depression, as well as another one will be under the Alzheimer's. So these are the something like the drug with like the extract which is having another pharmacological responses as well. And this is a oxalation. And by preliminary phytochemical screening, I could find that all the plants what I have selected, the majority constituents like alkaloids, flavonoids, terpenoids, saponins, anthraquinones, so which is majorly responsible in treating neurodegenerative diseases and responsible for the formation of neurogenesis, that is formation of new neurons. So all those constituents were present in the extract what I have selected. Oral acute toxicity studies I have done to know either this extract is favoring the animals to carry out the work or not, and I have not seen any morbidity and mortality after the 14 days of the inducing or giving the dosage of 2000 mg per kg body weight by selecting female mice. So I have selected the dosage as 400 mg.
Oh, PhD 1, only 2 minutes left.
Yeah, okay. So this is the thing how I have selected the methodology. So group 1 to group 8 and different dosage, inducing, etc. And these are my results where I have compared the other groups with the disease control on 0th day, 28th day, 56th day. This is aluminum chloride induced forced swim test. And these are the results for tail suspension test on 0th, 28th and 56th day. And this is for scopolamine induced 0th, 7th, 14th and 21st day. And this is for tail suspension test, again for scopolamine induced. So by following Dunnett's test, so where I can compare the multiple variables with the other groups as well. So disease control was compared with all the other groups, and I could say that this was comparable with the standard drugs whatever I have selected. And because of multiple constituents present in the plant extract, like it might be vitamins like ascorbic acid present in capsicum annum, thiamine is present. So in that way, number of vitamins as well as.
Yeah. Thank you, PhD 1. Now the session is open for Q&A.
Yeah, sure.
Yes, madam. Sudha ma'am, please proceed.
Yeah. Any specific active ingredient which you said, because first of all, it's a combination of 3 herbs, I understand. So
Yes,
any
yes.
specific active ingredient that you would say was responsible? Have you quantified
Yes,
each of the component?
Yes, ma'am, I have quantified. So one of the alkaloid content which is present in Tiliacora racemosa, that is bis-benzyl isoquinoline, which I found is one of the reason in treating depression in amnesia, ma'am.
So have you tried to isolate that and purify the compound?
Yes, ma'am, we can proceed with that. Like we can isolate it and we can go for further studies, ma'am, to know exactly what is the mechanism by this alkaloid. It has shown the responses, but further studies are needed for this to tell exactly to what extent it is able to treat the diseases, ma'am.
Since the theme of this research congress is with respect to innovation, so where would you like, what is your future scope of this particular research project? Can you?
Yes, ma'am. The future scope of this work will be like as the life expectancy nowadays, as due to the technology advancement, the life expectancy is increasing. And dementia is, Alzheimer's is one disease where the person after 65 years of age, the person is suffering with Alzheimer's. And as I mentioned, this is even cause depression as well. So by this study, if I can formulate a single drug which can be used to treat depression as well as dementia, it's not separately taking the drugs, and diagnosing in the earlier stages can be helpful, ma'am, so that this drug formulation can be helpful to some extent in treating both the diseases.
Thank you. Over to you, Gautam, sir.
Yeah, that might be the future.
Yeah, okay. So good, you
Yes.
have presented very well about the polyherbal mixture. So
Yeah, thank
my
you, sir.
question is, like, what motivated you to go with the polyherbal instead
Yes,
of
sir.
one or two, or separate one and then go with a particular molecule or alkaloids which can be useful for antidepressant plus dementia?
Yes, sir. Sir, already with a single individual drug, as I mentioned in the earlier of my slide, individually I have carried out on Teliacora resimosa, sir. One I have done, and another one is on fenugreek. Already I have done. So the work is even published in elsewhere also. So, sir, I wanted to know the synergistic effect of different extracts. Right, already two I have done, sir. I have selected another one as capsicum annum. So I wanted to study the synergistic effect of this three component, which has already shown or possessed a required um, a principle or mechanism to treat depression and Alzheimer's. So that motivated me to select and carry out my work by selecting these three different extracts.
But already
Yes.
the individual component, okay,
Yes,
that is well established. It is useful for either for antidepressant or for dementia.
Yes,
right?
sir.
Okay.
Yes, sir.
So
Yes.
going directly with the polyherbal extract to the animal, have you analyzed these things on in vitro through cell line, etc., whether the combination with this ratio would be useful or having the more synergistic action?
Sir, yeah, already like in vivo and in vitro method I have carried out, sir, but cell line I couldn't. Maybe, sir, there is a chances that this polyherbal may possess better responses by using some in vitro in silico methods also, after knowing the structure, etc., by either GCMS or docking, etc., sir.
Right, right, right. So
Yes,
it's
sir.
good work, but only a little bit if you add something in vitro testing here, which can
Yes, sir.
make your research more rational. Okay, like you are going
Yes,
step
sir.
by step. Additionally,
Yes,
or like individual component which are available in the market. Okay, you can
Yes,
take
sir.
that on extract and check with in vitro. If it is synergistic is coming, then you can select which out of these 3 or 4 polyherbals, which 2 or 3 are more efficient against these diseases.
Sure, sir. Sure, sir. Sir, I would like to say something. As you mentioned in vitro, I have carried out Elman's method, sir, like to know the inhibition property of the extract, where like based on different colors
Right, right.
on
Okay.
acetylcholinesterase enzyme. So I have carried out, sir, but I couldn't mention here because as they mentioned only 10 minutes. So
Yeah.
briefly, just I mentioned, sir.
So
That's
one
all.
more suggestion for the slide
Sir,
of presentation.
yes, sir.
Can you present
Yes,
the
sir.
results? Okay, go to your slide of results like the previous one. Previous, where the table is tabulated. So it's very difficult to anyone to read this individual value. Okay, if it is a
Yes,
bar
sir.
diagram or something which can concisely you can present it, it's very good for the audience. Okay,
Yes,
this
sir.
data is good for a thesis, good for the publication, but for presentation it should be very concise and very representative and expressive of your results or outcomes.
Okay, sir. Okay.
Okay, so this thing you can take care about your in future presentations. That is
Sure,
only my
sir.
suggestion
Sure,
about
sir.
it. Yeah, overall
Thank
very
you, sir.
good.
Thank you.
Thank you.
Yes, sir. Thank you, sir. Thank you.
Thank you.
Yes.
Most of the points have been covered by Dr. Gautam and the madam. So,
Yes,
as
sir.
rightly suggested, therapeutic positioning need to be addressed. What are the available therapies? How
Okay,
is
sir.
polyherbal? What is your thought process behind selecting all these combinations? So, therapeutic position is very, very important. And
Okay,
my
sir.
suggestion is, if you require, you can discuss with some doctors working on this field. They can
Okay,
also
sir.
add value. Okay. So that's
Okay,
all
sir.
from my side.
Okay, sir. Thank you, sir. Thank you, sir. I will surely follow the advice, sir.
Thank you, OPHD1.
Thank you, ma'am. Thank you.
The next presentation is by OPHD2. OPHD2. The last call for OPHD2. Okay, so we will be moving on to the next presentation. OPHD5. Next presentation is by OPHD5.
Hello. Hello.
Yes.
Hello.
Yes.
Yes, am I audible?
Yeah, you are audible.
Yes, I request team to kindly present my presentation because from yesterday I am facing the issue.
Okay. Ma'am will shortly present it.
Yeah, sure.
OPHD5, your presentation is visible now.
Yes, thank you
So,
so much.
okay, so you can start. You have 10 minutes.
Okay. Good afternoon to everyone. Today I am going to present on the topic of formulation, development and evaluation of ion activated in situ gel system using reverse phase Hplc method. Next slide, please. Contents of my presentation are introduction, aim and objective, experiment details, conclusion and references. Next, introduction. In this particular slide, next slide please. The ion triggered in situ gel system by using the single drug as well as the combination of the two drugs has been prepared in the literature by using the various polymer system such as sodium alginate and the gums to treat the various ocular disorders like bacterial infections, conjunctivitis and allergic reactions. Individual formulations patient has to administer for treating those type of the diseases. So to reduce the frequency of the formulation administration, only one formulation has been prepared, that is in situ gelling system, by using the two drugs ciprofloxacin hydrochloride and olopatadine hydrochloride, which are having the activity as antibiotic and antihistaminic respectively, So we have prepared here the one formulation containing the two drugs in combination for treating those diseases. Next. Hello. Hello. Am I audible?
Yes.
Will you please?
Okay. Yeah.
Yes.
It's
Next is aim and objective. So formulation, development and evaluation of ion trigger system by using the reverse phase HPLC method. This is the aim. Objectives of my work was to develop and evaluate ion trigger system by using the polymeric system, by using the gums, by using the two drugs, antibiotics and antihistaminics simultaneously, and to develop such a formulation which enhances the drug permeability, patient compliance and cost effectiveness. Next slide, please. Hello. Next is experiment details. In the experiment details, this is having the two steps. First is the development and the validation of the reverse phase HPLC method for the simultaneous equation of the two drugs. And second step involves the formulation development of the in situ gel system. For this, we have prepared the 9 different batches F1 to F9 by using the 3x2 factorial design. The APIs were selected with the preservative and buffering agent. It has been added in the polymeric solution like a gum. Here we have used the gums like xanthan gum, guar gum, and the galan gum. And based upon the optimization, we have selected here the galan gum. All the prepared formulation were evaluated for the physical evaluation such as the clarity, viscosity of the sole, gel, gelling time, pH, and the assay. All these three out of 9, 3 optimized formulations have been selected and studied for the stability study, excellent stability conditions, and effect of the sterilization. The excipient compatibility studies, drug content and the percent drug release was calculated with the or determined with the help of the reverse phase HPLC method, which is developed and validated. And lastly, we have selected the one optimized formulation from this and tested for the antimicrobial susceptibility testing by the agar-well technique or the couplet method, and the HChem study was done for the irritation study. Next slide, please. First, I am going to focus about the reverse phase HPLC analytical method development and the validation. In that, will you please go to the next slide? Next slide. Here we have tried the different trials, and based upon that, we have selected the optimized conditions. Next slide, please. Here we have selected the C18 Agilent Zorbex SBAQ column. Next slide, please. And we have selected the mobile phase system as the trifluoroacetic acid 0.1% and
I think slide is
40.
not moved. Yeah. No.
Yeah. Here we have selected the concentration of the drugs as 30 microgram per ml and 70 microgram per ml for ciprofloxacin hydrochloride and olopatadine hydrochloride respectively. We have selected here 260 nanometer as a selection of the analytical wavelength. 0.1% trifluoroacetic acid in water, 60% and 40% acetonitrile. 40% we have selected as a mobile phase system. Here we have used the C18 column. Injection volume is 10 microliter. Flow rate was 1 ml per minute, 70 minutes for zone time, and 30 degrees Celsius is the column temperature. Based upon all the trials, we have selected this optimized condition, and the graphs were shown. The summary of this, the developed method is being validated as per the ICH guidelines for the specificity by the identification, specificity by the force degradation study, stability of the analytical solution, intermediate precision, method precision, accuracy, linearity, robustness, ruggedness. So we have developed successfully the analytical method, reverse phase HPLC method, and validated the same. Next slide, please. Next, please. Next. Yes, next slide, please.
Yes. Can you just wait for 2 minutes?
Yeah.
This slide correct?
No, before that.
Okay, before that. Fine.
Formulation development of this. Before that also. This, yeah. After that, you can move to the next slide. Now I'm going to focus on the formulation development of the ion triggered in situ gel system. Next, please. Here we have used the different parameters. Please next. Yes. So here we have selected the drug gelling agent as the different combination or different ratio of the gums. Based upon the preliminary trials, we have selected here the Galan gum, as I have mentioned. Tonicity agent we have selected as the mannitol, buffering agent as the boric acid tromethamine, preservative as the BKC, and the vehicle is selected as the water, and simulated vehicle or the media is used as the ATF, having the pH 7.4. Next slide, please. Then on that basis, I have prepared the different formulation batches, F1 to F9. Next slide, please. By taking the different ratios of the polymeric system and as well as the excipients. Next, this is the formulation table of the same. Next, all these 9 formulations I have kept for the excipient compatibility study for the period of the 30 days at the accelerated temperature condition and the humidity condition, and analyzed by the reverse phase Hplc method. Next slide, please. And it has been assayed by the Hplc method.
You're still on slide number 13. Is it moving?
No, ma'am, it is not moving.
Sorry, ma'am, it's not moving due to some network issues. We are trying.
Okay. Okay.
This is the excipient compatibility study. It's chromatograms and the assay. And here we have found the assay between the 98 to 10% for the ciprofloxacin hydrochloride, and for the olopatine hydrochloride 98 to 101%. Next slide, please. Ma'am, may I just explain the next part?
Please go ahead. Next,
in the second evaluation, we have done the physical evaluation of all these 9 different batches by clarity, viscosity of the solution, viscosity of the gel, and the gelling time, and the pH of the solution. On that basis, based upon the viscosity of the sol and the viscosity of the gel, we have found the F1 to F3. It has shown the viscosity of the gel 10 times to that of the viscosity of the sol. Gelling time we have also found consistent, and the pH of the solution was in the range as desirable for the ocular system. So on that basis, out of the 9 formulations, we have selected the 3, that is F1, F2, and F3. Please move to the next. All these 3 formulations we have optimized for its assay by the same reverse phase Hplc method which has been developed. Next slide, please. And found the percent assay of the formulation F2 around 98.62.
OPHD 5. Last 1 minute left.
Then after that, the accelerated stability study is also been done. Then effect of sterilization of the pre-sterilization and post-sterilization has been done. On the basis of this, F2 formulation is in vitro study is being carried out and found it is following the Crossmire-Pappas release model. Preservative study of the benzalkonium chloride by the same method has been developed and analyzed. Antimicrobial susceptibility study is being done by the agar well diffusion method by using the different strains as mentioned in the reference books. And by performing the headchem study, the irritation score was also been found around 0.18 for our formulation. So it has been found non-irritant. So I can conclude that the reverse Hplc method for the simultaneous estimation of the both the drug has been developed and validated successfully, and the formulation of the in situ gelling system were prepared and evaluated successfully.
OPHD 5, your time is over.
Yes, thank you so much.
The session is now open for Q&A.
Dr. Gautam, you can start.
Yeah, thank you. Yeah, it's good presentation and few basic questions, few basic suggestion also first. Just I want to know what was your priority. Okay, when we present anything, is it your preference or priority HPLC method development or formulation development?
Sir, in this particular case, my priority is both.
Both. See, you are either working in PhD, having the objective, major objective. Okay. If someone is working in analytical method development, then maybe the major objective is to development of the analytical method. So you are working in area of formulation or analytical?
Sir, I am working in both.
Both.
First, I have developed, the combination of the two, because the so my core was both.
So there should be a sequence of incidents. Okay, like if you are saying it is the first time you are going for this formulation, so obviously formulation objective is first. Okay, and the background, how you came with this, why it is novelty in this formulation, that we should express first. Okay, now to analyze or determine the drug in the formulation, then there is a need of the method development.
Okay.
Okay, so you should present in that way the objective of this one formulation development. In that, the one part is what? There is an analytical method also, because there is no such method is reported. So far, you are developing for that method also. Okay, so that, because somewhere it is a confusing whether you have expressed first analytical method, then you are saying you prepare the formulation. Okay, anyway, but you can think of it. You have sufficient time right now. Okay, so while improving your work or compiling work, make a predefined objective and sequence for your work presentation. Okay.
Okay, sir. Thank you.
So my next question is a very basic question to you. Why you have not developed the UV method? Why only HPLC method?
Sir, in this particular case, in the next part, I'm going to do the same thing, but because it is the PhD work, the UV is not as much as accurate as that of the HPLC, so I have moved towards that.
So only that reason, like UV is not accurate, that's why you have not developed the UV?
Sir, I am planning for the same, because the UV method is also not available.
So see, there is another, we can say our suggestion, or like you say, when we start PhD, we should think about from the fundamental thing. Okay, first UV. Okay, if it is UV is available, can we explore information from UV? If it is a UV is not there, if it is required, for what purpose UV is required, and what purpose you are going for HPLC directly. Okay,
yes, sir.
So there is certain reasons for why HPLC method is required. Can you explain?
Yes, basically, first I have chosen the two different drug combination. One was not working, one was not having the UV absorbers. It is not UV active substance. So I have tried with the same combo, and then later on I have changed my drug, and then the combination which is having the UV absorbance and showing the similar, showing the peaks, or it is UV active. So I have moved towards the HPLC.
So that is another story you have added. Okay, what drug you have selected before and not, it doesn't matter here. Okay, now you have to discuss whatever the drug you have selected, you have prepared the formulation, you have developed the method. So these two drugs, okay, generally if you go for HPLC, then what is the reason? What are the drugs? Is there a method for the single analyte is available or not?
Yes, it is available for the single drugs.
Okay, then what is the combination which is not there?
Yes.
Okay, so in formulation, why this combination is required? What is the rationale for these two drugs?
The rationale behind these two drugs is to treat the bacterial infection or the antibiotic, to get the antibiotic activity as well as the conjunctivitis activity by taking the antihistaminic drug in the combination with the antibiotic. So this is not.
Is there any preclinical studies are reported this two drug combination would be suitable for that?
Hello.
Is there any preclinical study or any literature reported for this two drug combination? Because there is a n number of combinations are there for anti-inflammatory or this thing. So why these two drugs only? Okay.
Sir, actually,
yeah, please go ahead.
This olopatadine hydrochloride, that is the anti-conjunctivitis drug or antihistaminic drug. In N-Pharm, I have worked on the same, so I was having the lots of literature about the same. So based upon that, I have selected the another thing, which is there, the antibiotic. So the combination was not available, and in one or the two or the more of the literatures to treat the different diseases, some drawbacks was there. So I have chosen this particular combination.
Right. Okay. Your thought process, like little bit, we need to think about whether the mechanism or what is the incidence of these two diseases, what is the incidence level or prevalence of these two diseases altogether. Okay, what is the any other combination with the same categories are available or not. Okay, so if those things are there in that, then it is, you can easily justify your combination also, because this would be obvious question for till your PhD examination. Everyone will ask why you have selected these two combination only. Okay, and beforehand, generally we do the in vitro testing whether they have the synergistic action or only 1 + 1 only, or 1 + 1 is equal to 5. Is there any synergistic action is there or not? So certain preliminary, though you have not done, but you should have a certain literature which indicates that yes, these two combinations would give a better outcomes.
Sir, actually I have done, but I have represented only the few things here.
Right. Okay. So you should be in a concise manner. You should also express, because these are the questions I asked, because it's a quite obvious question. So you should think about the audience point of view or the examination point of view. Like beforehand, you should also express something about this thing in your presentation also. Okay. So next, my question is like the viscosity which you have generated here, the formulation which 9 trials are there. What are the variables you have optimized here?
In 9 different formulation batches, I have checked the clarity of the particular solution, viscosity of the sole, viscosity of the gel, gelling time, pH, surface tension, as well as
characterization. No, this is characterization. In formulation variables, which component you try to optimize in the 9 trials?
I have tried to optimize the polymer concentration.
Okay, so only the polymer concentration, on what basis? Only clarity and viscosity, is it that, or pH characteristics, etc.?
Yeah, based upon all these parameters.
So again, challenge is like, see, only gelling agent concentration in a PhD, that would not be sufficient. Okay, you think about it. What other variables are there which you can optimize it? Okay, with respect to formulation component, with respect to the process component also. When you are saying the two objectives are there, method development and formulation development. So as a formulation expert, I will say it's 9 trial and formulation is optimized. Okay, in a PhD work, it's there is many more things to be optimized.
Sir, actually, I have applied the QBD in the same. I have taken the critical quality attributes, QTPP, process variables, excipients, and the manufacturing related process. All those has been also considered into the consideration.
But when I see your presentation, I did not find anything. Okay, my question is definitely from your presentation only. So as a presenter, you should give what you have done very concisely. Okay, so that the examiner will say, okay, yes, you have done so far, and from that you reach to these 9 trials. Okay, which QBD, which DOE you have applied for your formulation optimization?
3/2 factorial design I have applied. 3/2 factorial design.
3/2. Okay, what is 3 and what is 2 here?
3 is nothing but the formulation batches, and the 2 is the variables which we have selected.
So 2 variables and 3 is what?
3 is the formulation batches. 3/2. So 9 different formulations we have prepared. Variable
3
and the
levels. 3. You mean 3 levels, right?
Yeah. Levels and 2 factors.
Two factors. Yeah. So
dependent and independent variables on that basis.
Which are independent variable here?
Here, independent variables were the concentration of the different excipients, like the which are not going to affect.
Thank you. That's all. Yeah, thank you. That's all from my side. Yeah, madam, you please proceed.
Thank you, Dr. Gautam. Somewhat my suggestions are in line what sir said. So I won't repeat them. But just talking about your title, and my thoughts are in concurrence with Dr. Gautam sir, that you wanted to focus on method development or a formulation development. That clarity should be there with respect to the title of your presentation. Then secondly, in the order in which you are presenting, your compatibility study should always be before formulation development. Your pre-formulation study cannot be, you did these formulations and then you are showing excipient compatibility. That doesn't happen. Okay. So most of the points which I wanted to cover, sir has already covered. But just one question from my side. If at all, if you want to scale it up, what will be your plan with respect to manufacturing? What will be your order of addition and how you would do that?
First related to this, the solution is going to be prepared by taking the drugs, by taking the preservatives, by taking the other excipients. First will be that. Second will be the solution of the polymer and polymeric solution, and then this will be the second, and then we are going to add the first into the second, and then first we are going to identify the sterilization effect on the same, and then the post sterilization effect as well, and then we are going to mix on the same.
Okay, thank you. That's it from my side. Over to you, sir. Dr. Mishra.
Hi, Arun here. So if you can note down the suggestions given, so next time, all the research fellows will be benefited. So in the first slide, there should be a scheme of research. In this scheme of research, you can say that out of 10 items listed in the scheme of research chart decision tree, I will present only this 1 or 2 part in this presentation. Okay, forum will be aware of that you have done so much work now because of limited time you are presenting this. Again, it may be possible that during your final PhD examination, some ophthalmologist may review your PhD presentation. So my suggestion is that if you have discussed with some ophthalmologist, that is good. If you have not, then you should consult to understand the practical implication of this one. Okay. Now, third point will be that there are formulations in the market. Have you done any comparison study with the marketed formulation?
Yes, sir. In the market. Hello. Yes, sir.
Yeah.
In the marketed formulation, there is in situ gel was not available. So we have tried a level best to procure the innovator's product, but still the formulation which is available separately, like both the drugs individually. So we have done the same also. In vitro study, I have done by taking the eye drops.
So you need to be careful in selection of this, and somewhere you can keep in the backup slide that you have done so work with the marketed formulation. So that's all from my side. Thank you.
Yes, sir. Thank you so much.
Thank you, OPHD 5.
Thank you so much.
We'll be moving on to the next presentation by OPHD 6.
Yes, I'm there. Am I audible?
Yeah, OPHD 6 is audible. Can you please switch on your camera?
Yes. Is the screen visible?
Yes, it's visible.
Are the slides moving?
Yes, they are moving.
Okay. Should I start?
Yes, you will be getting 10 minutes.
Yeah, thank you. Good afternoon, respected judges. Today I am going to present my research work entitled In Silico Approach for Development of Hybrid of 1,3,4-oxidiazole chalcone derivatives as EGFR inhibitor. Abstract number is OPHD 6. These are the contents of my presentation, starting from background to references. It is the journey of research in the field of treatment of cancer. Cancer, though but currently sidelined by COVID-19, it will definitely cause havoc throughout the world within next few years. Cancer is the disease where certain cells in body multiply uncontrollably. According to WHO statistics, the cancer deaths are rising, and in 2020, around 10 million deaths occurred worldwide, and it's the second leading cause of death. The cancer can be treated with the help of various anti-cancer drugs. The anti-cancer drugs act on targets of the cancer cells, which are known as receptors. Some of that kind of receptor, which is recently emerged as a better target, is epidermal growth factor receptor. It is having a central role in cancer growth, and it's a cell surface receptor which contains two parts, the outer part and the inner part. My research is focused on this inner part of the receptor, which contains a tyrosine kinase domain, which is the target for my research work. The drugs act on this tyrosine kinase domain, and they inhibit the cell proliferation and signal transduction process. There are these drugs are known as tyrosine kinase inhibitors. Up till now, there are 4 generations of tyrosine kinase inhibitors are discovered. But these tyrosine kinase inhibitors have an issue of drug resistance. To manage this drug resistance, the patient need to be administered with the different anti-cancer drugs which are acting on multiple targets. But that leads to an pill burden in the patient. So this issue of current tyrosine kinase inhibitor of drug resistance and pill burden can be overcome by combining 2 scaffolds in a single molecule. These scaffolds can be linked together by a linker, and the molecule that will become is known as hybrid molecule, and the mechanism is known as hybridization. These hybrid molecules have some advantages like increased specificity, better patient compliance, it can overcome pharmacokinetic drawbacks, fewer adverse effect it can show, and a reduction in chemo resistance is seen. This motivated me to work on hybridization approach. So aim of my study is to design, synthesize, and anti-cancer activity of hybrid molecules for managing drug resistance and pill burden. The first and the very crucial step of my research was selection of nucleus. The proven facts related to 1,3,4-oxidiazole nucleus and the chalcone have influenced my decision to use them. Rationale behind this work is there is one drug which is in the clinical trials currently, which is containing 1,3,4-oxidiazole nucleus, which is known as zibotentan, which shows an anti-cancer activity. There are 2 drugs which contains chalcone nucleus like butene and isoliquiritingenin, which are standard drugs available in the market which combat the EGFR inhibition. So I have taken these 2 scaffolds together, and they are connected with an amide linker, and that makes a hybrid pharmacophore that I have taken for my study. Description of work, the component of the work was carried out using computational modeling, which is also known as in silico studies. The in silico study was started with QSAR studies, then designing of hundreds of NCs, then ADME, docking, and molecular simulation. The lead molecules which were obtained in silico studies were further taken for synthesis in laboratory, and its confirmation is done using spectral studies. These are the different softwares which were used for in silico studies. A thorough literature survey was first carried out on 1,3,4-oxidiazole and chalcone nucleus as a EGFR inhibitor, and this is the summary of the substitution that can be made on these 2 pharmacophores, and this helped me to design the molecules. This is the series that I have taken for QSCR studies. It contains 24 molecules. This is the result of 3D QSCR studies that I have carried out using VLife MDS software. A statistically significant model 1 was generated, and it helped me to know what substitutions to be made on the pharmacophore that I have selected. These are the 3D plot which are generated, which help me to know the substitution pattern. So this is an outcome of QSCR and SAR studies that I have carried out through literature and the actual in silico studies. So this helped me to know what substitutions to be made so that I'll get an effective EGFR inhibitory activity. So based on this outcome, I have designed almost around hundreds of molecules, and that I have passed it through Combilib tool of VLife MDS software. And this is the outcome of Combilib tool, where you can see here 27 molecules come out of those 100 molecules which have shown screen score of 5 and 6 and with a good predicted activity. So these molecules I have taken further for molecular docking, which shows here the table is displayed, which shows a binding affinity and number of hydrogen bonds. Total 13 molecules have shown the better binding affinity as compared to standard, and compound number 97A had shown an highest binding affinity when I docked these molecules in the crystal structure of EGFR using PDB ID 1M17. This is the picture of compound 97A with a crystal structure of 1M17. This is for standard, and these are the key highlights of the docking studies, which shows around 50% of molecules that I have taken for study have shown a key binding with an amino acid met 769, similar to that of standard, that assured me that the docking has carried out in a correct pocket. The molecules were further subjected to ADME studies using Swiss ADME's online tool, which is available on Google. That helped me to know the drug likeness of the molecules. So among 13 molecules, there were 9 molecules which were obtained, which are having good drug likeness property and good toxicity profile. The toxicity profile is checked with the help of PKCSM tool. There is a simulation study is also carried out on the lead molecule, the molecule number 97A, which gave me an assurance that the whatever dock complex is formed, it is having a stable complex when it goes into in the living cell. This is the RMSD plot obtained. This is the 2D interaction plot and binding energy obtained with the molecule number 97A. You can see here MET 769 is showing and key binding with this compound. This is the scheme of synthesis that I am following. There are 6 steps. First 3 steps are for oxidizole preparation, 2 steps. Next 2 steps are for chalcone, and the last step is where hybrid of oxidizole and chalcone is prepared. For synthesis, I have used a green chemistry technique. Microwave synthesis I have tried. These are the compounds which are in the pipeline. I have
2 minutes left. Remaining
with only last step where the hybrid molecule is formed. Oxidizole and chalcone preparation is over. I think this is the representative intermediate IR spectra of one of the oxidizole. The spectral studies were carried out on IR, NMR and mass for the intermediates. This is the outcome. In nutshell, I can say I have taken a hybrid of 134 oxidizole chalcone, hundreds numbers that filtered through Combilib, got 27 molecules. They are further docked, got 13 molecules. They are further passed through ADMET filter, where I have got 9 molecules and 1 molecule was tested for simulation study, and the lead molecules are under synthesis process. Relevance of study, this in silico study and green chemistry approach used are saving on time, money and resources. So definitely it helps in environmental preservation. The hybridization approach which is used in this case will definitely have and get a single hybrid molecule which can able to manage drug resistance and pill burden in the patient, cancer patient. The future plan, I will be doing the cytotoxicity or MTT or SRB assay. Then in vitro EGFR assay will be performed. And I will be trying the action of these NCs on SRC and ILE, that is interleukin assay, so that its activity on multiple target can be proven. These are the publications that I have done through this work. This is a review paper which is published in 3.8 impact factor, Web of Science and Scopus Index Journal, mini reviews in medicinal chemistry. This is the research publication that I have done in Research in Drug Design and Discovery. Again, it's a Bentham publication, impact factor 1.15. And this publication is in pipeline, which is communicated to Elsewhere Journal, Journal of Indian Chemical Society, impact factor 0.2. These are the key references. I would like to conclude in the words of famous research scholar Mark Pattison: research is always incomplete. Thank you for your patience listening.
Okay. Now the session is open for Q&A.
Yeah, Dr. Gautam.
Yeah, thank you, madam. Yeah, overall, it's a very good presentation, and I just want to share that all of the students, those who are pursuing the PhD and are the participant for this conference, they should learn from.
Sir, we can't hear you. You're muted.
Yeah. Am I audible now?
Yes, sir.
Yeah, I was saying every participant of this conference should learn from this presentation that how well we can present our objectives, research outcomes, future plans. So that is the good learning. I really found this presentation skills is very good with this candidate. Okay. And it's in a very good language, layman language. Okay, even though I'm not very in depth, I'm knowing about the synthesis, etc. But I understood what she is going to carry and how I can correlate some other allied areas with this work. So it's good presentations, first of all, and congratulations that many things, outcomes you have presented as a publication also. That is a requirement for any PhD scholar to bring outcomes from their work at every step. Okay, so she has not completed her work, but she has brought outcomes from whatever the work she has done so far. It's good work. Okay.
Thank you.
Yeah. So I appreciate, and all of us should we learn from the everyone. So this is a good learning we can see from her. So my questions to about the technical point of view. Okay. When you synthesize these molecules, okay, and ultimately you want this molecule should come to the market and should translate to the patient. Okay. Other than the once you have discovered or you have optimized the lead and you are going for the further activity, what about the certain other physicochemical properties which you will look into the molecule so that it can easily translate to the patient's hand?
First, I need to check on the its physicochemical parameters like its lipophilicity, aqueous solubility, so that the formulation can be done in a proper way and it can be administered by patient effectively, and it should reach to the target also. So I should look after those parameters, and I have performed in silico studies over that. So very soon, once I get those compound, I can try out some laboratory work on that, so that I'll have some proofs with me.
Okay. So thank you. That's all from my side. Okay. I request other judges to please, madam, please proceed.
Yeah. Thank you, sir. In line with the question that was asked by Gautam sir, I would like to take that question ahead. What I understand that you have not done right now at this stage adequate pre-formulation studies for your molecule, but based on whatever literature as well as on your in silico study that you have done, what do you anticipate for your molecule? That's one question from my side. And what properties are you predicting, anticipating? So with that, what will be your approach for taking this drug ahead? And number two, what will be your approach to right now? How much quantity have you synthesized? You have synthesized this molecule, madam?
Yeah, I have synthesized intermediate compounds, and one of the hybrid molecule I have completed. Synthesis of hybrid molecule I have
So
completed.
what is the scale? How much quantity you have synthesized right now?
It's around 1 gram, 1.5 grams are with me for one hybrid molecule.
So imagine that if you want to scale it up, what will be your strategy if you want to take this ahead with respect to bulk manufacture and process technology? So what will be your strategy? These are my two questions.
Okay. The first question was related to consideration in terms of pre-formulation studies. Are you asking, ma'am?
Yes, with respect to properties of your molecule, because it's not just you synthesize the molecule. The practical, the properties of the molecule also qualify it whether it can go ahead with respect to, from bench to bed. So what properties do you foresee for your molecule based on whatever data that you have?
Actually, I have performed some in silico studies wherein drug likeness is already checked. So probably the compounds, when I perform those study in laboratory, then these compounds probably will show a better drug likeness properties also. And in terms of formulation, if I talk about, I should be able to see whether which kind of dosage form is possible for these molecules to be converted into some of the dosage forms, so that it can be easily taken by the patients. And the second question is related to I have done a pilot study wherein a microwave synthesis is used. Since the microwave synthesis is used, it has not taken much time to synthesize a hybrid molecule. So on bulk, if I'm talking, that may not be a much costly process. It must be a cost-effective process, since I have already screened out some molecules from 100 numbers which I have designed. So there is a cost reduction is definitely is possible as compared to conventional methods that can be followed for synthesis. So that is the answer of second question.
Yeah, that's it from my side. Dr. Mishra?
Yeah, thank you. And this is a very nice presentation. And in earlier presentation, we were discussing about the schema of work, decision tree, something like that. So in this presentation, you have covered all the points. Okay.
Thanks.
And so I'm in agreement with the comments given by Dr. Gautam and Madam. So all the best. Thank you.
Thank you, OPHD 6. Next presentation is by OPHD 7.
Hello.
Yes, you are audible.
Yes. Is my presentation visible?
Yes, it is visible.
Okay. Shall I start?
Yes, you can start.
Okay.
You'll be getting 10 minutes.
Okay, thank you. So, good afternoon, everyone. The title of my presentation is In Vitro Studies of Functionalized Hologram Nanoparticles for Therapy of Glioblastoma. This will be the content of my presentation, where I will be talking about introduction, rationale, aims, objectives, synthesis, optimization, and characterization of hologram nanoparticles, drug loading, release studies, in vitro studies, and summary. Introduction. So basically, according to National Brain Tumor Society, there are many different types of brain tumors, of which the tumors of astrocytes, that is astrocytoma, is the most common brain tumor. WHO grade 4 astrocytoma, which is glioblastoma, is one of the most aggressive forms of brain tumor. The problem with this disease is the survival rate is very less. It is less than 10% in case of patients. The current diagnosis includes use of conventional MRI and functional MRI along with MRI spectroscopy. The treatment is a mixture of a combination of rather surgery, radiation, and chemotherapy. Surgery is basically done to debulk the tumor to remove excessive dead tissues, which is often followed by radiation. Radiation to remove any residual cancer cells if present. This may or may not be supplemented with chemotherapy, wherein temozolomide is the standard drug used. Now, the conventional therapy presents itself with many challenges. One is the presence of blood-brain barrier and blood-brain tumor barrier. Also, the physical chemical properties of the drug that are used also limit the penetration of the drugs to a few millimeters. Another is tumor heterogeneity, which is in the target tumor site, there is a mixture of tumor cells as well as healthy cells. So it becomes very difficult to localize your drugs there. Another is that with long-term exposure of temozolomide, there are some residual cells which convert themselves into glioblastoma initiating cells, and they function as cancer stem cells. So these cells are responsible for drug resistance because they have many deregulated DNA repair pathways. So that's why we propose to use glutathione functionalized hollow-gold nanoparticles with dual drug loaded, that is temozolomide and resveratrol loaded hollow-gold nanoparticles for therapy of glioblastoma. Now the question is, why hollow-gold nanoparticles? Is because hollow-gold nanoparticles have some unique properties, like their shape and size is easily tunable. They have good surface functionalization techniques. For diagnostic purpose, they can be used as fluorescent and luminescent agents. They are biocompatible in nature. Given their size, they function on the principle of enhanced permeation and retention. That is, they can easily be permeated and be retained in the target tumor tissue. Also, the surface of these particles show resonance. They show surface plasmon resonance, which can be made use for photothermal therapy with using an external NIR laser. The purpose of using two drugs, that is using resveratrol along with temozolomide, is that since I previously mentioned that temozolomide is prone to resistance, resveratrol is known to enhance, basically known to sensitize the residual cells to the action of temozolomide. It also causes suppression of the MGMT enzyme, which is responsible for temozolomide resistance. The reason for using glutathione as an active ligand is that in case of glioblastoma, all these cells show overexpressed glutathione receptors. So that's why we are using glutathione for active targeting. And photothermal, I think someone is, yeah, okay. And the release of the drugs will be potentiated with using photothermal therapy. This is because since I mentioned that hollow-gold nanoparticles have a good SPR, they have good ability to convert the photothermal energy to the required heat at the tumor site, which will help the particles to penetrate and accumulate better. These are my aim and objective, which is to develop functionalized hollow-gold nanoparticles for therapy of glioblastoma. These are my objectives. The first step is synthesis. So we have used sacrificial galvanic replacement method for synthesis of cobalt nanoparticles. There are two steps involved. First is to synthesize a core nanoparticle, which is a cobalt nanoparticle, and on the cobalt we will be depositing the gold layer. So in case of synthesis of cobalt nanoparticles, these are the 5 factors which are very important. There are 3 things that are used in synthesis: trisodium citrate, which is used as a stabilizing agent, cobalt chloride salt for making the cobalt nanoparticle, and sodium borohydride as a reducing agent. So the ratio, concentration, and sequence of addition of these 3 are critically involved. Another is use of citric acid. Citric acid can be used to potentiate the stabilization of sodium citrate, but we have synthesized batches without citric acid as well. The reaction time before addition of sodium borohydride and the hydrolysis time after the addition of the sodium borohydride. So after optimization, we have found out that when sodium borohydride is added 2 minutes after addition of cobalt chloride and sodium citrate, And when the hydrolysis time is kept up to 45 minutes, we were able to synthesize particles with the SPR 808 nanometers. Also, we need to protect the entire process against action of oxygen. Another is gold shell growth. In the case of gold shell growth, the rate and amount of addition of cobalt nanoparticles from the particles to the gold is very important. The concentration of gold is important. The stirring time is important. PVP can be used as a stabilizing agent, but it has been noticed that PVP dysregulates the surface of the particles, and hence the functionalization of the active ligand will not be possible. Coming to characterization of hollow-gold nanoparticles, uh, so we have characterized the particles for SPR, that is surface plasmon resonance, using UV spectrophotometer. The morphology was studied using HRTEM. Here we can see that the hollowness is visible and there is a layer of gold on the outside. The particle size and zeta potential were analyzed using Malvin zeta sizer. The average particle size was found to be 84 nanometers and zeta potential was minus 17. To stabilize nanoparticles, we have functionalized the nanoparticles with pegylation, and then we have conjugated the glutathione. On the left side, there is an image of before and after conjugation. There is a slight increase in the actually there's a significant increase in the size of the particles after pegylation and GSH conjugation. We have used a modified PEG. The one with the PEG has two terminals. The SH terminal binds to the gold and the COH terminal binds to the glutathione. So basically the PEG will also be acting like a bridge between the glutathione conjugation and the hollow gold nanoparticles. Here the first FTIR is of glutathione and the second FTIR is of the particles functionalized with glutathione.
You have 2 minutes left.
Yeah. Coming to the photothermal heating of the hollow gold nanoparticles, the particles are irradiated with NIR 808, and we have seen that 40 degrees heating is seen within 350 seconds. That is almost around 5 minutes. The particles reach the temperature of 40 degrees. The entrapment efficiency of both the drugs, that is temozolomide, was found to be 50% and resveratrol was around 41%. The release was studied using NIR laser 808 with a power of 3.2 watts. It was studied in phosphate buffer 7.4 and 95% of temozolomide released within 4 hours, and resveratrol was around 80%. Coming to the in vitro studies, the first is cytotoxicity studies using MTT assay, where we have seen the cytotoxicity effect of the drugs on various glioblastoma cell lines C6, LN229 and U87. In the first study, we have seen whether temozolomide and resveratrol were capable of showing a synergistic effect, and we found that since the IC50 was lesser than the individual drugs, therefore it was more cytotoxic. Next is cytotoxicity studies of using the laser. So we can here see that with using the laser, the IC50 of the particles has IC50 has decreased. That suggests that it gives a better cytotoxicity. And lastly, we've loaded individual drugs in the hollow gold nanoparticles and combination. Here the combination has shown the IC50 of 115, which is drastically less than individually loaded hollow gold nanoparticles. So there shows some synergism. Then we carried cell cycle analysis, and since temozolomide and resveratrol both show a rest in the S and the G2 phase, all the individual drugs and the hollow gold nanoparticles have arrested the cell growth in the synthesis phase, that is the S phase of cell cycle. And lastly, yeah, I'll just wrap up. And lastly, we have done the apoptosis studies, which give the percentage of the drugs that are viable apoptotic in the late and the early phase, and the amount of the drug that had undergone necrosis. The results suggested that all the particles killed the cells via late or early apoptosis. So here I am concluding that from the in vitro release studies and the cell culture studies, dual drug-loaded hollow gold nanoparticles can have shown promising results and can be used further for in vivo evaluation as well. Thank you.
Now the session is open for Q&A.
Yeah, over to you, sir.
Thank you, madam. Yeah, it's good presentation.
Good afternoon, sir. Sorry,
Yeah, please,
sorry.
Arun sir, please go ahead. Please go ahead. Please go ahead.
Sorry to interrupt. Just one thing. We are tight on schedule. So and we have I guess 6 more presentation left. So just just to remind you guys so we are tight on time. Thank you very much.
Thank you, sir. Yeah, Arun sir, please go ahead.
Yeah.
Please
So one thing I suggest the improvement in almost all the presentation that we need to present the competition scenario, because whatever work we are doing, that is for the benefit of the population and for the industry. Okay, we want to commercialize our research in the form of product or something like that. So we need to present what are the competition and where our product will fit into.
Okay.
It is very much important. So that is the end goal. Correct. This is from my side. Thank you.
Okay. Thank you. Yeah, Gautam sir.
Yeah. Thank you. Dr. Arun has mentioned clearly. Okay, and before explanation, you should say what are challenges and where your product will meet the requirement or fulfill the gap. Okay, that should be addressed. Yeah. Okay. And my question, like you have prepared this, loaded the temozolomide drug in that. Okay.
Yes.
And there is a possibility that temozolomide will immediately convert into its metabolites. Okay. So how will you ensure that it will not convert while the circulation in the blood circulation, it will not convert into its metabolite?
Okay. So for that, sir, actually this is a targeted therapy. So this will be given via the IV route, and as soon as the drug will, within the fraction of seconds, the NIR laser will be targeted at the target site. And when the drug releases, obviously it will convert it into its metabolite for the activity. But since it is an IV route, so there will be a very less, it will not stay in the blood circulation in the surrounding tissues for lot. It will directly go to the brain, to the tumor cells.
But obviously, have you checked it immediately after IV? It is whole drug is available to the brain. It is in any case, it is not possible. But whatever you give through IV,
correct.
Even a fraction of only few drug or like a fraction of drug only it will go, and there is a rate of going or passing through the BBB and it will reach to the target site.
Correct.
So I suggest like you should also check while circulation time how much drug is releasing. Okay, which is converting it to MIT or ATC its metabolite from TNZ. So this will help you like what dosing you are targeting. From that, how much it is reaching to the brain.
Sure. Okay, I will surely take your point into account, and also I'll just mention that the release of the drug is only when the NIR laser is focused on the nanoparticles, because the NIR laser will be responsible for the physical desorption of these drug from the surface of the hollow-gold nanoparticles. So since we'll be focusing the laser only on the tumor cells, so basically only...
Right, okay, but no study is revealing that the drug is not releasing in the blood pH.
Yes, so we are actually in the process of the in vivo studies. I've just
that's why obviously this question will come to the exam and we have not showed that one. Yeah,
correct.
That's it from my side. Okay, thank you. Madam, please proceed.
Yeah, good presentation. Objectives well explained. Just one question from my side is, you have first pegylated it and then conjugated it with glutathione.
Yes.
Have you checked what is the objective of first pegylating it and then glutathione conjugation?
Okay.
Have you seen what happens if you reverse the order?
Yes, ma'am. So basically, we are the pegylation steps involves the stirring of the nanoparticles for 8 to 10 hours. So when first the drug was loaded and then it was pegylated, the drug leached out. So less drug entrapment was noticed, but when also pegylation helps to stabilize the hollow-gold nanoparticles. Without pegylation, the hollow-gold nanoparticles are stable for a period of 8 days, but with pegylation, they are stable for 3 months and more. So that is why doing the pegylation step gave good entrapment results.
But should you not first conjugate and then pegylate? Because what I understand is pegylation is more with respect to improving the biological half-life of NP. Because you are giving it by IV.
Correct.
So first step, line of defense is to pegylate to improve the biological half-life. And then when the drug has reached the brain, the role of glutathione will start.
Okay.
So should you not think of reversing the order?
Sure, I will look into it. Yes, ma'am, sure.
Okay, thank you.
Yeah, thank you.
Yeah, thank you. Yeah, this is really good presentation, and you have presented very well, and all the graphics, etc. you have covered. It's really good, appreciated. Yeah.
Yes, thank you, sir.
Thank you, OPHD 7. And the next presentation is by OPHD 3.
Thank you for welcoming, ma'am. Hear my voice, ma'am? Shall I continue?
Yes, you are audible.
Ma'am, yes.
Yeah, please go ahead.
Yes, good afternoon. Yeah, yes, ma'am. Thank you. Good afternoon, one and all present here. Thank you for giving this best opportunity to all the organizer and the team members. Thank you.
Presentation is not visible.
Yeah, ma'am. Few seconds, ma'am.
Okay.
Ma'am, my screen is visible, ma'am?
Yes, it is visible.
Yeah. Thank you, ma'am. Thank you. Shall I continue, ma'am? Shall I start, ma'am?
Can you make it a little bigger? Because I think you are presenting from mobile, madam.
Yes, ma'am. Unfortunately, my laptop is not working, ma'am. That's why I changed the...
Can organizers present on madam's behalf? Yeah, now it's visible. Better.
Ma'am, shall I proceed, ma'am?
Please go ahead.
Yeah, thank you, ma'am. Thank you. So, good afternoon, one and all present here. My topic, the assessment of green synthesized cerium oxide nanoparticles from peel extract. Actually, the peel, that is the Punica granatum peel, will be selected. That is the plant will be, because of why I am select the Punica granatum peel, that is the pomegranate, means it is a bio-based product. The layer, particularly the layer, it will be utilized for the synthesis of nanoparticles, especially the cerium oxide nanoparticles. Most of them, the cerium oxide nanoparticles, it will be rarely the synthesized in the green pigment, that is the green synthesized product. So first I will select the Punica granite peel. Then after the analysis of the green Punica granite peel, next will synthesis process will be start. So before starting the cerium oxide nanoparticle synthesis process, I will share about the first of all, everything you know that that is the nanotechnology. It is useful for a wide variety of areas. That is the synthesis, characterization, and also the explorations, applications of nanosynthesized materials. So biotechnology is said to be the it will be useful for in the various biological applications. So they have everyone know that they are having the many type of nanoparticles is that is the silver, gold nanoparticles, zinc oxide nanoparticles. I will choose that the cerium oxide nanoparticles. It is the trivalent and the tetravalent oxidation state of cerium oxide nanoparticles. It is having the many activities, especially antioxidant, antibacterial, and antifungal activity. It is one of the simplest work. Initially, they having the doing the punica granatum peel, it will be useful for the productions of cerium oxide nanoparticles. So after the synthesis of cerium oxide nanoparticles, I point out and the result, getting the results from the anti-fungal activity. So these are having the many activity, especially I report that antifungal activity using the 2 microorganisms. So the cerium oxide nanoparticles have been synthesized in the punica granatum peel extract. So once they are synthesized in the punica granatum peel extract of cerium oxide nanoparticle, I confirmed with the Raman spectrum, that is the characterization studies I confirmed. So these are the peel, the collections, the peel, it will be, first I collect the fruits, then remove the peel. After drying that peel, next it will be formed into the powder form. By using only for the mixtures, that is the collections and preparations of the sample. Once I will be separate the powder, productions of powder. Next I will move on to the cerium oxide nanoparticles. How to they are productions? I referred to previous articles. So how do they are synthesize the cerium oxide nanoparticles? So first they are ethanol water extractions of nanoparticles. So first the 10 gram of peel powder, they are added into the 100 ml of double distilled water in the particular degree Celsius. After that is the 15 minutes, they are stirred continuously 80 degrees Celsius. Then after the centrifugation process, they are separations of pellet and the supernatant. The pellet powder, it will be fully dried with the help of the incubator. After the incubations of pellet, they are formed into the yellow color powder. So finally, I conclude that is the cerium oxide nanoparticles. Once the powder will be formed with the help of these cerium oxide nanoparticles. So only one thing. So mostly they have the 3 way, that is the physical and chemical and biological. So the chemical method, the cerium oxide nanoparticle, cerium nitrate hexahydrate act as the chemical reagents to adding this product. That is the first, initially the peel powder, it will be mixed with the cerium hexanitrate product will be utilized. Then finally it will be conformed with the cerium oxide nanoparticles. So the characterization studies, the Raman spectrum. So the Raman spectrum work is the Raman impact. It is a powerful detection technique. So mostly already everyone know that is the UV, FTER, EDAX, and initially the Raman spectrum, it will be get the peak and accurate value. So that's why I will select this Raman spectrum, because of once they are confirmed means next it will be move on to the cytotoxicity or genotoxicity studies. So the Raman spectrum, the peak value at 452 centimeter power minus 1. These are my graphical abstract. That is the showing the peak value of that particular cerium oxide nanoparticles. So they are confirmed with the previous studies. So already they are analysis the previous studies. They are peak value the 450 to 500. So that's why I confirmed these are the cerium oxide nanoparticles. Once I confirmed these cerium oxide nanoparticles with the help of characterization, next we'll move on to that antibacterial and antifungal and antioxidant activity following the DPP, HSA, all the things. But now I report that antifungal activity Aspergillus niger and Candida albicans. So these are the results. How do they are the fungal activity will be present? So positive control amphiotensin B, it will be used. Once the sample cerium oxide nanoparticles, the ranges 13 mm ranges will be inhibitions activity will be present, and also the similar to the Candida albicans, that is the 14 mm ranges of cerium oxide nanoparticles will be present. Both of them, the positive control amphiotensin B will be used. So finally, what I actually this is for simple work. Once they are cerium oxide nanoparticles will be synthesized from the punica granatum peel. So after the synthesis of that is the nanoparticles, I confirmed with the Raman spectrum, and also finally I determined they having the antifungal activity or not. So they are used into 2 ways. Thank you, Ananda.
Arun here. It was a nice presentation. So how will you ensure the consistency of this punica granatum?
Sir, pardon, sir. Can't hear, sir.
Different varieties of punica granatum depending upon the region and all this?
A red color pigment, red color.
No, fine. But I am saying, are there different varieties of this punica granutum. How will you
Yes.
ensure that punica granutum, which has been collected in the north or south or east or west, are same? Characterization and all this. Are you getting my question?
Yes, yes, sir.
See, whatever work you have done, that you have taken some, whatever was available with you. But how will you ensure the consistency in that particular punica granutum? How will you characterize that?
Actually, the peak value, sir. That is the peak value I will determine that the punica granatum peel having the cerium oxide nanoparticles.
So, have you checked with the punica granatum from other part of the country or something like that? Is there same?
Yes, sir.
Okay, thank you.
Thank you, sir.
Gautam,
Thank
sir?
you. Yeah.
Yeah.
Dr. Gautam.
Yeah. My
Thank you,
question,
sir.
have you prepared this extract, and then from that extract you prepared the nanoparticle?
Yes,
So,
yes,
what is
sir.
the need to prepare the nanoparticle, and what is the expected to be delivered through oral, topical, or through IV? In what way it is thought to prepare a nanoparticle for this extract?
Yes, sir. Actually, the chemical preparation by using the cerium nitrate hexahydrate, that is the chemical. It will be, and also they having the cerium chloride, all the things. But this peel powder, it will be mixed with the cerium nitrate hexahydrate. It will be finally the reporting cerium oxide will be present. That's why I confirmed with the, and also peak value will be analysis for that is the cerium oxide nanoparticles.
That is fine. My question is, what is the need, what is the rationale for making a nanoparticle formulation, whether in what is the intended delivery route for the extract or these particles? Am I
This
clear?
form he's asking, to be precise.
What was the need? What was the necessity to make nanoparticles if you had other better options?
No, ma'am. Actually, it is will be rare. The synthesis of cerium oxide nanoparticles when compared to other nanoparticles. That's why I will be synthesizing this cerium oxide nanoparticles.
Okay. Once we prepared, okay. Next questions. What you expect? How it can be delivered to the patient or to the fungal disease, through oral, through topical? In what way it can be given?
Powder, in case it is a medicines formation, sir. Actually, these are having the antifungal activity means they are given to next step cytotoxicity or genotoxicity activity will be present means it acts as a medicines. I give the powder.
Okay, thank you. That's all from my side.
Just one simple question, very quick. What will be the cost of your product? You are making it as a nanoparticle. What do you think would be the cost of your product? And is it industry scalable?
No idea, ma'am. Sorry, ma'am.
Okay, thank you.
Thank you, ma'am. Thank you. Thank you for giving me this opportunity. Thank you.
Thank you, OPHD 3. The next presentation would be by OPHD 8.
Hello.
Yes.
Am I audible?
Yes, you're audible.
Okay.
OPHD 3, please stop the screen sharing. Okay, fine. Yes.
Shall I stop?
No. The presentation is visible and
yeah, you can start your audible as well.
Okay.
Go ahead.
Good afternoon, judges. My topic of presentation is novel ligand conjugated albumin nanoparticles for the co-delivery of hydrophilic and hydrophobic chemotherapeutic agents for synergistic breast cancer therapy. This is the flow of my presentation. So first start with introduction and hypothesis. So as we know, breast cancer is the most commonly diagnosed malignancy and leading cause of mortality among women worldwide. So among all the available treatment options, chemotherapy is the primary treatment option for the management of breast cancer. So the combination of chemotherapeutic agent is the most viable option as it overcomes the chances of multi-drug resistance, increase the therapeutic efficacy by targeting multiple pathways of the cell cycle, and it also decreases the systemic toxicity due to dose reduction. So the gemcitabine and docetaxel combination is well tolerated and used as a first-line therapy for the treatment of several solid tumors, including breast cancer. However, their co-delivery is associated with certain limitations as gemcitabine is highly hydrophilic drug with a short plasma half-life. It undergoes rapid decomposition into its inactive metabolite in the plasma by cytidine deaminase enzyme. So we need repeated dosing to maintain its therapeutic level, which leads to the systemic toxicity. Whereas the docetaxel is a BCS class 4 drug with poor solubility and low permeability, and we need co-solvent and surfactant mixture to dissolve the drug, which leads to several anaphylactic and hypersensitivity reactions. Therefore, in order to overcome the above drawbacks, here we have hypothesized to prepare anacardic acid, gem conjugated, bovine serum albumin conjugate loaded with the docetaxel drug. So anacardic acid was chosen as it is a natural fatty acid analog having anti-cancer properties, and it is reported to target the tumor cells via its binding affinity to the VEGF receptors. And the docetaxel drug was physically loaded into the bovine serum albumin due to its high protein binding affinity. So albumin has been chosen as a carrier first reason due to the drug has high protein binding affinity. Second, it is also reported to internalize into the cells via SPARC and GP60 receptors. So upon IV administration, they will nanoparticles will enter the tumor cells or tumor microenvironment via APR effect, and then they enter the tumor cell through ligand receptor interactions and reach the site of action to exert their therapeutic activity. So first of all, the synthesis of anacardic acid, BSA gemcitabine conjugate. So here we have utilized the free amino and carboxylic group of the bovine serum albumin to conjugate anacardic acid and gemcitabine. Firstly, the carboxylic group of anacardic acid was activated using EDC NHS as activating agent and was made to react with the amino group of the bovine serum albumin. Secondly, the carboxylic group of the albumin was activated with EDC and NHS and was made to react with the amino group of the gemcitabine, which results in the formation of conjugate. Further, the conjugate was characterized by Mardy-Toff and gel electrophoresis assay. So here we have shown the molecular weight of native BSA molecule. So the center of mass distribution of native BSA molecule is around 66,206. Whether upon conjugation of the anacardic acid and gemcitabine, the molecular weight shifted to 71,218. So this confirmed the formation of conjugate. In line with the Mardy-Toff results, the gel electrophoresis assay also revealed the increase in molecular weight of the conjugate as compared to the native BSA molecule. Then the conformational changes of the bovine serum albumin molecule was analyzed using CD spectroscopy. So as here clearly visible, the anacardic acid gem modified BSA conjugate showed significant conformational changes as compared to the native BSA molecule, though it has been reported in the literature that these conformational changes further improve the tumor cell uptake of the albumin nanoparticle via interaction to the GP16, 18 and GP30 receptors. Then the prepared conjugate was utilized for the preparation of nanoparticles. So nanoparticles were prepared by high pressure homogenization process and was characterized for various critical quality attributes. So the pH of the albumin solution was maintained 8. The particle size of docetaxel loaded anacardic acid modified nanoparticle was 163 nanometer with the PDI of 0.13. The SEM and AFM images confirm the spherical and smooth surface of the prepared nanoparticles. Secondly, the in vitro release study of the docetaxel was conducted in two different pH conditions, pH 5.5 and pH 7.4, and it was found that the docetaxel showed biphasic release profile with immediate burst effect up to 4 hours and then sustained release up to 48 hours. The second image shows the plasma stability profile of the gemcitabine conjugate. So here, as clearly visible, the free gemcitabine showed around 64% of the inactive metabolite content, whereas it was only 16% in case of conjugated gemcitabine upon 24-hour incubation in the plasma. The in vitro hydrolysis study of the gemcitabine conjugate was done in pH 7.4 and 5.5 in the presence or absence of protease enzyme, as protease is known to cleave the amide bond, and it was found that the gemcitabine release was higher in acidic pH, which was further increased in the presence of protease enzyme. Then the cellular uptake, as we know, docetaxel will act at cellular level and gemcitabine site of action is nucleus. So the cellular uptake and nuclear co-localization assay was performed using coumarin C6 as a fluorescent dye. So using two cancer cell lines, MDMB231 and MCF7 cell line. So as shown here, the anacardic acid gemcitabine BSA loaded coumarin 6 shows higher green fluorescence intensity as compared to the native BSA nanoparticle in both the cell line. We showed the effective internalization of nanoparticles inside the tumor cells. The overlapped image, that is panel E, which is the overlapped image of the panel A and B. Panel A is the blue staining dye DAPI which stained the nucleus. So this panel E and panel F, that is box analysis, showed the effective co-localization of the C6 loaded anacardic acid nanocarrier in the nucleus as well as cytoplasm, whereas in case of native BSA nanoparticle, there was negligible green fluorescent intensity in the nuclear region. And the apoptotic assay was done for all the formulation and free drugs to assess its therapeutic potential. So as shown here, the docetaxel loaded anacardic acid modified BSA nanoparticle showed higher apoptotic index as compared to the free drugs and their combination. Then in vitro cell cytotoxicity study was conducted in MDMB and MCF7 breast cancer cell lines. So as shown here, the docetaxel loaded formulation showed higher concentration as well as time dependent cytotoxicity as compared to free drug and their combination, which could be due to the effective internalization of this nanocarrier as compared to free drugs. Then pharmacokinetic analysis was done after IV administration of the nanoparticles and was compared with the marketed formulation Taxotere, which is docetaxel marketed formulation, and Gemzar, which is gemcitabine marketed formulation, and various pharmacokinetic parameters were calculated. So the AUC and T half of docetaxel was 6.12 and 6.28 fold higher as compared to its marketed formulation, whereas for gemcitabine, the AUC and T half was found to be 3.27 and 8.9 fold higher as compared to its marketed formulation.
PhD 8, you have 2 minutes left.
Okay. So then pharmacodynamic study was conducted in DMB-induced breast cancer model. So as shown here in figure A and B, the docetaxel loaded anacardic acid treated group showed significant reduction in the tumor burden as compared to the marketed formulation and their combination. These are the photographic images showing the tumor which were isolated from the respective treated group. So here also clearly visible the significant reduction in tumor size of the docetaxel loaded BSA nanoparticles. Then toxicity study was conducted by estimating the level of various liver as well as kidney toxicity markers such as aspartate transaminase, alanine transaminase, malonyldehyde, blood urea nitrogen and creatinine, and it was found that there was no significant change was observed in the docetaxel treated anacardic acid BSA nanoparticles in all the toxicity markers as compared to control group, whereas other formulation treated groups showed elevated expression of all these markers showing their hepatotoxic and nephrotoxic nature. Then in vivo hemolytic toxicity was checked by analyzing the RBCs via SEM, and it was found that docetaxel loaded nanoparticles showed no visual changes in the RBC structure as compared to control group, whereas in marketed formulation treated group there was severe structural and shape deformities of the RBCs. So conclusion, these anacardic acid functionalized nanoparticles showed sustained drug release profile, improved plasma stability of the gemcitabine, promoted higher cellular as well as nuclear uptake of the drug, and higher apoptotic index as compared to free drugs. The developed nanoparticles in vivo showed significant improvement in the PK as well as PD therapeutic profile, and also resulted in reduction of the toxicity which are associated with the marketed formulations. So the fruitful findings of this study can offer an additional research horizon for drug macromolecule conjugate to overcome the limitations of hydrophilic and hydrophilic drug molecules. Thank you.
The session is now open for Q&A.
Yeah, Gautam here. Yeah.
Yes.
Yeah, I have just quick questions. Like you mentioned in the rationale that this combination is expected to overcome the multi-drug resistance, right?
Yes, sir.
So what is MDR?
Okay, so multi-drug basically is of two types, acquired resistance or the intrinsic resistance. So acquired resistance, like we say, when it develop resistance, the drug will not show efficacy. So as docetaxel.
Got it. Okay, thank you. Now, in your case, have you done any study where you can claim this thing? Yes, it can overcome the MDR. Because in your cell line, etc., nowhere I found that you have evaluated this can be utilized for the MDR.
Actually, sir, there is no such study has been performed because our motive was to just to get the synergistic efficacy.
Okay.
But in this case, as we have taken the MDMB 231 cell lines, so it already overexpressed several multi-drug resistance transporters such as PGP. So we have got the improved cellular uptake in case of nanoparticles. So from that we can justify that yes, and even the cytotoxicity results, we have got the synergistic cytotoxicity in the cell lines. So
good,
we can.
Good. So you should know like which data to represent your claim also. Okay, that is the only point I raise here. Okay, it's good work and very well presented. Okay, and you have explained the results in a very concise manner.
Hello.
And it's a very great work we have done from the
Sir,
starting.
you are not audible.
Am I audible?
Yes, sir, you are
Hello.
audible.
Yeah, okay, thank you. So it's a really good work. So from the beginning to the end, till the pharmacodynamic study, you have performed very well. The last questions, like what you feel the translational approach or limitation in this case?
Sir, can you repeat the question?
What you feel, what are the challenges, okay, which are there from translate from lab to market?
Okay, sir. So first challenge will come in the synthesis of the conjugate. As we have synthesized it only in lab scale, that also 500 mg conjugate. So first challenge will be the reproducibility of the this conjugation synthesis. Second, we have used high pressure homogenization process for the synthesis of nanoparticle. So it is already a scalable technology and widely used by industry. So during preparation, there will be like we have optimized did the formulation up to 100 mg batch. So when we have to scale up, we have to see all the critical process variables as well as critical material attribute that can affect the critical quality attributes such as size, PDI, drug loading, etc.
Okay, thank you. That's all from my side. Madam, and please proceed.
Dr. Mishra?
So, if you refer your slide number 9 in the PK study slide, so have you done this work or you have gathered some these data from the literature?
Sir, actually your voice is not coming properly. Can you please repeat the question?
PK study 9. Am I audible?
Yes, sir.
Am I audible? Okay.
Yes, sir, you are
Yes,
audible.
sir.
So in slide number 9. So in slide number 9, PK study, have you taken this data from the available literature or you have generated this data?
No, sir, we have performed this study, pharmacokinetic studies.
So this was the only question. Fantastic work, good, very good presentation. It's right from start to end and a very logical conclusion. All the best. Thank you.
Okay, thank you, sir.
So let me congratulate you. D8, right? PhD 8.
Yes,
So the concept was very well presented. The work is complete in all sense. Just one question from my side that here, as Gautam sir said and Mishra sir said, that here you are establishing synergism and also you are trying to overcome MDR, right? So
Yes, ma'am.
what is the, do you see any reduction with respect, based on your findings, do you see any reduction with respect to dose requirement when you are using those two molecules in combination?
Okay, ma'am. Actually, here the combination index was not calculated because already this combination is well reported in literature that it exhibits synergism. So we have not calculated that combination index, and we didn't check the dose reduction index for the drugs. But as we have done the MTT assay, there we have found that compared to the single drug treatments, the combination has showed the significant reduction in the IC50 values. That dose reduction index we have not calculated because we have took the combination from literature as it shows synergism, so we did not perform the study.
And which animal model did you use?
A DMBA induced breast cancer model.
Okay, thank you.
Yeah, just I want to add to OPHD 8. You can take the suggestion of Sujata ma'am. Okay, like you just check it how synergistic. Okay, how you can minimize the dose. That would be further application of your research work, because you have done very good work. So further a small study will value add in your work.
Okay, sir. I'll surely do that.
Yeah, thank you.
Okay, sir. Thank you, judges.
Thank you, OPHD 8. The next presentation will be by OPHD 9. OPHD 9. OPHD 9, your presentation is visible. Can you please on your camera? OPHD 9, can you please switch on your camera?
Am I visible?
And you are not audible as well. Okay, you are now visible.
Am I audible?
Yes, you are audible. You can start the presentation. You have 10 minutes.
There is slide is visible, ma'am. Can you confirm?
Yes, slide is visible. You can on the slideshow.
Yes. Thank you for the confirmation. Good afternoon. Greetings and warm wishes to all. My abstract
Can you be a
is
little bit loud? PhD 9, can you be a little bit loud?
Yes, yes, okay. Good afternoon. Greetings and warm wishes to all. My abstract ID is OPHD9 and the title of presentation is Qualitative Design Optimized Cooking Model Liposomes for Treatment of Breast Cancer using Photodynamic Therapy. Let's see the slide one by one. The flow of presentation is introduction about cancer and breast cancer and photodynamic therapy and liposomes.
PhD 9, your slide is not moving.
You're still on slide number 1, the title slide.
Okay, from my side it already moved. No?
Please on the slideshow.
It is in slideshow mode.
Is it possible organizers can present on his behalf?
Yes, ma'am, we can. Okay, now it is moving. Now you are on the slide 2.
Maybe network issue.
Can you take it on slide presentation mode, a little bigger? Because it's still... take it on full screen mode, please.
Why? It is on full screen mode.
Here it doesn't seem to be on full screen mode.
Organizer Vaishnavi, you can present on behalf of him.
Yeah, I think, yeah, Vaishnavi, can you present it on his behalf?
Yes, ma'am, we can.
Yeah.
Is the presentation visible?
Yeah, it is visible.
Yes,
Yeah,
yes.
visible.
Okay. Okay, so OPHD 9 can start with the presentation.
Okay, thank you. Good afternoon, greetings and warm wishes to all. My presentation ID is OPHD9. The title of presentation is Quality by Design Optimized Curcumin Nodal Liposomes for Treatment of Breast Cancer using Photodynamic Therapy. Next slide. The flow of presentation is introduction about cancer and liposomes, aim and objectives of the present study, methodology adopted for the preparation of liposomes, result and discussion, conclusion, and future perspectives of the present study. Next. Coming to introduction, cancer is the abnormal growth of cells in the body. Generally, cancer cells grow. Proliferation is more than normal. Generally, cancer cells proliferate compared to normal than normal. Generally, cancer cells proliferate much faster than normal cells. The mortality of different cancers around the globe was more than 10 million in the last year. More specifically, breast cancer caused 2.26 million deaths in the females. Different strategies like chemotherapy, targeted drug therapy, and photodynamic therapy were attempted to treat breast cancer. Next slide. Among the photodynamic therapy recently received more attention due to painless and non-invasive approach to treat breast cancer. PDT involves light, photosensitizer, and oxygen to induce oxidative stress by enhancing reactive oxygen species to induce cancer cell death. Curcumin is a potent water-soluble herbal drug present in the form of powder in yellow color and capable of generating ROS upon application of light. Thus, liposomes could be a potential strategy to overcome the poor water solubility and improve the therapeutic efficacy after loading into liposomes. Coming to aim and objectives, the aim of the present study is development of curcumin nodal liposomes using quality by design approach for treatment of breast cancer with the application of photodynamic therapy. To achieve the above aim, following objectives are framed to prepare stable curcumin nodal liposomes using quality by design approach, to characterize and evaluate curcumin nodal liposomes, to perform the safety study of curcumin nodal liposomes on healthy cell lines, and to perform the anti-cancer activity study of curcumin nodal liposomes on breast cancer cell lines. The methodology adopted for preparation of liposome is thin film hydration method. Curcumin and lipids such as soybean phosphatidylcholine and DSPV PEG 2000 and cholesterol have been used to prepare the curcumin nodal liposomes. The solvent chloroform was used to dissolve the lipids and drug. Then further the solvent was evaporated and the thin film is formed. Further the hydration was used using pH 7.4 phosphate buffer saline. Then the formulation was sonicated and stored in refrigerator. Coming to result and discussion, the formulation was optimized using the quality by design approach. So based on thorough literature, quality target product profile. The formulation was optimized using quality by design approach. So based on thorough literature, quality target product profile for curcumin nodal liposome has been selected for therapeutic efficacy. Selective cytotoxicity to breast cancer cell line, nanocarriers like liposome, particle size should be less than 200 nanometer, drug and drop and drug loading should be high, release profile will be sustained release, and the ROS generation of formulation should be maximum, and it should be on high cytotoxicity to cancer cell, and it should not affect the normal healthy cell lines. Next. After getting process parameters, formulation parameters, and raw material evaluation methods, fishbone diagram has been drawn for further studies. Next. The Watson design was used to prepare and optimize curcumin nodal liposomes. Independent variables like amount of SPC and amount of cholesterol and hydrogen time have been selected, and the dependent variables like particle size, entrapment efficiency, and drug loading.
OPHD 9, you are not audible. OPHD 1, you can unmute. Please unmute.
Is he audible?
No, madam. He's
No,
not
ma'am.
audible.
Ma'am, actually his mic is on mute. OPHD 9, can you please unmute? You are not audible. Okay, yeah.
Watson design was used to optimize the curcumin nodal liposomes. Independent variables such as amount of SPC, amount of cholesterol, and hydrogen time were used. But the dependent variables like particle size, entrapment efficiency, and drug loading are selected. After doing experimentation, and the data has been plotted into a design expert software, and the software suggests the optimized formulation for preparation of curcumin nodal liposomes based on the particle size. The model suggested quadratic model, and the entrapment efficiency and drug loading also. The model suggests quadratic model based on the R square and based on predicted and adjusted R square. The difference between predicted and adjusted R square was less than 0.2. Thus, the model was significant, and the lack of it was insignificant. Next slide. Next slide. The software suggested various effector variables and various responses for optimization of curcumin nodal liposome. Based on the equation, the positive simple shows are directly proportional to the.
OPHD 9, you have 2 minutes left.
Based on particle size, the positive symbol shows are directly proportional to the raw materials, and the negative symbol shows the indirect proportional to the raw materials. So particle size showed increase in concentration, SPC shows increase in particle size. Increase in cholesterol concentration shows a decrease in the entrapment efficiency and drug loading. Next slide. The optimized formulation showed 120 nanometers, and the opacity potential was -8, and the scanning electron microscope showed spherical shape of particles, and there is no drug crystal was found. Then optimized formulation entrapment efficiency was 66%. The drug loading was 1.81%. Next slide. The FTR study showed that curcumin showed 3500 OH group, and the aromatics showed 3010. The Planck liposome showed its characteristic of SPC and cholesterol, and the drug order formulation showed that curcumin peak was absent. It might be due to entrapment of curcumin in the formulation. Next slide. Next slide. Curcumin showed the characteristic of 180 degrees Celsius, but in the drug order formulation D, it was absent. It might be due to non-crystalline and amorphous conversion of curcumin into formulation. Next slide. Next slide. Next slide, please. Before one slide will be there. Before that. Before one slide, please. No, yeah.
Have you discussed the results of your cell line studies?
No. Before one slide. Previous slide. Go to 15, 15, 14.
14th slide.
the formulation has showed sustained release of drug release formulation, and the drug release kinetic study showed that maximum oxide value found in viable model, the beta value was found 1.25. It showed that complex release mechanism, initial sustained and initial burst release and later sustained release. Next slide.
I go.
The ROA studies of healthy cell like HEK293 cells and MC7 cell breast cancer cell lines were used. The application of light was based on absorption of curcumin, blue light. Curcumin absorbs at blue light region. The light used for present studies 460 nanometer. It was found that the ROA generation is increased with increasing concentration of curcumin. The site is in this MC7 cell lines also the ROA generation was increased within
10 minutes are over. The session is open for Q&A.
Yeah, Madam Gautam here. Yeah. Am I audible to you?
Yes, sir. Yes.
PhD 9?
Yes, sir. Yes.
Yeah. So you have considered this BBD, box blanket design for a formulation optimization, right?
Yes.
So what is the rationale for selection for BBD instead of other surface methodology?
Because generally it is widely used and it is useful for 3 or more variables.
But there is also CCD is also there, central composite.
Yes.
So what is the, yeah.
But it is generally easy and run also only 15 runs have been used for the preparation of formulations, preparation and optimization.
Okay, but hardly few more will be there in CCD. But what is the rationale for selecting the BBD? Anyway, okay. So you have screened certain material attributes and process parameters,
yes.
Right? So which risk assessment you have performed for identify these variables?
Yes, sir. A preliminary study has been done based on different lipids we have used. At that time, we have screened initial particle size and stability also. Based on that, we have selected a species. So I have been postulated.
Fine. So I would like to suggest like you can add certain risk assessment tools about the failure mode or relative risk, which will more rationalize or justify your variable which you have taken for optimization. Okay, that will value add in your work.
Okay.
Okay. Okay, that's all from my side. Yeah.
Arun here. What challenges do you foresee while doing the scale up this technology or larger sector? What challenges do you foresee? Do you have any idea about that?
Sorry, your voice is not clear. Can you repeat, sir? Sir, can you repeat? Your voice was not audible.
What was the best that you have taken for this process? I said, do you understand? What was the best?
What was the
best size? Best size. Now, right? In the scale up of this technology, what challenges do you foresee?
During the scale up, the particle size will affect, sir. Depends on liquid concentration, depends on vessel size and sonication parameters. Purely, it will affect the scale up. And also stability of liposomes also it's also one concern for the scale up.
Okay, so how do you have any idea how?
Yes,
that is good that you know the problem. What the solution problem? So you need to work upon what are the critical factors, what are the CPC and CMA.
Okay.
By doing that, the thought of. Thank you.
Hello.
Hello.
Yeah, am I audible?
Yes, ma'am.
PhD 9.
Yes, ma'am. Yes,
Can you highlight the novelty of your project?
Yes, ma'am. In this study, we have used both red light and blue light for the anti-cancer to detect, to identify the anti-cancer activity of in the breast cancer cell lines. We have prepared the formulation and we used two different light, blue light and red light. Here, red light data was not shown because it was not in some literature. They showed that in curcumin is not stable. Due to the instability, it will convert into another bioactive product. So by using red light also, it will show the some effect. But in our study, we found that red light was not that much showed in that much anti-cancer activity was found compared to blue light, because blue light in curcumin showed maximum observance at 424 nanometer. We have generally it has broad absorption spectrum in the 420 to 480 nanometer. So we use blue light to detect the to identify assess the anti-cancer activity.
Secondly, just a suggestion: when you are presenting your data, you have presented particle size and zeta potential. Can you go to that slide, please? Yeah. So when you the same slide number 11. Slide number 11, please. So when you're presenting the data, just one caution: it appears as if you have cropped the image.
Yes,
okay. You must be having those parameters. Probably must have done on Malvern or any suitable analytical instrument. It appears that you have cropped that image. So whenever you're presenting that data, ensure that you give all those details, the entire parameters. Okay.
During image preparation, I made like for comparing these three images. That's why.
No, but whenever you are presenting it in any forum, or even you are presenting in any manuscript, refrain from cropping the image and showing it to the reader or to the audience.
Okay.
Just one last quick question from my side. Can you justify why did you use liposomes as a carrier over other carriers that are quite robust and stable?
Because liposomes are lipid-based drug delivery system, so cells can easily uptake the liposomes. So when it delivered with a drug, so cells can be easily engulfed and it will show the activity compared to other polymeric or other nanocarriers.
Okay, thank you.
Thank you, OPHD 9. The next presentation is by OPHD 10.
Hello, ma'am.
Yes, you are audible.
Okay, ma'am. Is my slide is visible?
Yes, your slide is visible and you can start with the presentation.
Okay, ma'am. Thank you so much. Good afternoon, everyone. Today I am here to deliver an oral presentation entitled Exploration of Selective Laser Sintering, that is SLS mediated 3D printing technology for rapid prototyping of solid oral dosage form. And as we all know that we are here today to discuss innovations in healthcare, then my work will mainly focus on novel and modern fabrication technology for solid oral dosage form. It will not mainly focus on formulation, but it will be a method specific or method focused work. Then coming towards flow of my presentation. Then I want to introduce all of you about selective laser sintering, that is SLS. SLS is a 3D printing technology, which is one of the powder bed fusion technology, in which we can use a thermoplastic polymer as a feedstock material with the help of heat energy produced by the laser source, this powder will partially
PhD 10, you are not audible.
made, then fused together.
PhD 10,
Which be
you can off your camera as you are not so audible.
yes, ma'am.
Can you please continue with slide number 3? We did not hear. Can you repeat slide number 3?
Okay, ma'am. Slide number 3. Am I audible now?
Yeah, you are audible now.
Yes, ma'am. This selective laser sintering is one of the additive manufacturing technology or 3D printing technology, which utilizes the heat generated by a laser source for a partial melting and then fusion of the particles to prepare any 3D object based on our provided CAD design. It will build any object based on layer by layer fabrication or manufacturing. Thus, we have used this modern manufacturing technology for fabrication of solid oral dosage form, where laser is used as a source of heat. Coming towards requirements of SLS, where we only required a thermoplastic polymer and a laser absorbing agent to prepare any object with the help of this selective laser sintering. Then it offers various process parameters like feed bed temperature, print bed temperature, laser power ratio, scan speed, hatch spacing, and layer thickness. Then the question is, why SLS? SLS is one of the modern and novel additive manufacturing technology which are having various advantages over conventional manufacturing of solid oral dosage form as well as any other 3D printing technology. Because in conventional manufacturing of solid oral dosage form, we have lots of steps like wet granulation, dye granulation, followed by compression or direct compression, which involve various processes and various excipients like lubricant, glidants, etc., etc. And in case of other 3D printing or additive manufacturing technology like fused deposition modeling or inkjet, binder jet, or any semi-solid extrusion, we have to prepare that feedstock. Means we have to prepare filaments or that inks or resins or particular gels for that fabrication. But SLS, the beauty of SLS is it is only single step procedure. No solvents are required, no additional excipients are required. We can mention this as a click and print method, where we have to just add our excipients in the feedstock. We have to give our CAD design and material will be printed as per our requirement. Using this technology, we can vary the shape, size, and dose of particular solid oral dosage form as per requirement of any pediatric, geriatric patients as per their requirement. Thus, it is highly personalized and customizable. Variation in all these process parameters will offer us the ability to play with internal microstructure, density, porosity of any solid oral dosage form. Thus, we can control the release of the drug incorporated in that. Thus, this is the aim of my study, and we have fulfilled these objectives. Then coming towards experimental design, where for the first time in the pharmaceutical field, we have explored the SLS 3D printer equipped with IR laser having 808 nanometer wavelength. Thus, using this printer, first of all, we took Colico tire as a thermoplastic material. We mixed air absorbing laser dye into various concentration in it. Then this blend we have prepared and we have analyzed for its physical and chemical stability by using physical and chemical compatibility by using DSC and FTIR techniques. Then this blend we have filled in the feed bed of our 3D printer. Then we have prepared a CAD design of a tablet having 10 mm diameter and 3.6 mm thickness. Then we inserted this CAD sketch or file in .stl format in our printer. We selected various process parameters as per our requirement and we started the printing. Thus, in the final step, we got the well-formed 3D printlets, which we have analyzed and characterized with various physical and analytical sophisticated instruments, which are mentioned here. These are the materials and methods which I have already discussed in my previous slides. CAD sketches I have discussed. So coming towards the
OPHD 10, you are not audible.
Thus, here we kept rest of the parameter constant and we selected one dye concentration at which it is printed. Then in the next series of formulation, we have used as it is that much dye concentration and we played with feedback temperature and print bed temperature. Thus, in the second step, we optimized the feedback temperature and print bed temperature, and followed by in the third step, we prepared series of formulation having various laser power ratios, and here we kept dye concentration and feedback temperature, print bed temperature as previously optimized. So this is the short overview of the process which we have followed to convert a virtual prototype into a physical prototype, which can be seen here. The effect of dye concentration where and a very low concentration of the feedstock material was not able to get print well. Thus, we increased it up to 1.25% concentration. So it showed the evidence of sintering. Thus, we considered it as the optimum concentration for the printing. This concentration we have used and we played with the feedback temperature and print bed temperature. Thus, it can be seen that at feedback temperature 130 degrees and print bed temperature 150 degrees, we obtained well-formed printlets. Similarly, at 130 degree feedback temperature, 150 degree print bed temperature, 1.25% dye concentration, we played with laser power ratios and we have obtained 3 such batches, L2, L2.5 and L3. Thus, in the next step, we characterize these printlets based on their physical dimension and weight variation, where we observed that closeness between virtual dimension and physical prototype dimensions getting more close with the virtual diameter and virtual thickness at higher feedback temperature and higher print bed temperature. Similar in case of a base.
You have 2 minutes left.
Okay, ma'am. When we increase feedback temperature and print bed temperature, closeness of virtual weight and physical prototype weight also increases. Coming towards L series optimization, characterization, here we found that closeness between virtual dimensions and physical dimension is obtained at laser power 3 specifically. Then weight also it is matching with the virtual dimensions in case of laser power 3, and laser power 3 is also giving maximum printing yield. Thus, we have selected the fabricated printlets at laser power 3 for further evaluation with sophisticated instruments. This is how we got to know the virtual weight of our printlets before its printing using mass property function of SolidWorks software. Then we evaluated a laser power variation batches with their dye content, breaking force, friability and disintegration time. Then we found that as we increase laser power ratio, dye content is decreased because maximum utilization of dye was there. Breaking force increases, friability decreases and disintegration time increases because as we increase laser power ratio, compaction, consolidation and degree of fusion of the particles increased. This is the thermal analysis, which is showing physical compatibility. This is the functional group analysis using FTIR, which is showing chemical compatibility of our used materials. In PXRD study, we can see that the semi-crystalline nature of KIR is preserved after getting exposed to higher concentration and high temperature and higher laser power ratios also. These are the actual images of same where we can actually experience how sintering is takes place, means fusion of the particle. We can see here the larger globules are the sintered or fused particles and the smaller
PhD
particles present.
Time is over. Time is over. 10 minutes are completed. And
these are the microcity images.
So yes, the session is now open for the Q&A.
Yes, ma'am.
I want to understand what is the how this technology will benefit how this will be beneficial.
Sir, we are in comparison with conventional manufacturing, we are reducing the time required, we are reducing the step, and we are reducing the excipients required. This is the one thing. And second thing is it is highly customizable and personalized. Thus, as per requirement, we can change the shape of our formulation, size of our formulation, and most specifically, we can also play with dose. For some genetically disabled
Correct.
person, need to give.
Yes. Sorry, but in generic, see, Indian pharmaceutical industry is mostly in the generic segment. Okay, herein we have to match with the innovator or marketed product. Okay.
Yes, sir.
Regarding, you have to keep more or less same weight, more or less same excipient as the innovator has taken, and you have to qualify the B study. Then only your be it NDA or whatever it is, regulatory agency will approve your product. Otherwise, it will go into the 505 B2 or something like that. So that's why I wanted to understand the objective of this work.
Sir,
regarding the in one of the sentence you have mentioned that that you are going to optimize the dose. Okay. So
Yes,
if you are changing the dose, then we have to do the clinical trials and all these things. And in generic pharmaceutical industry, time is the essence actually. So yeah, now you can say, you can answer.
Sir, this is the 3D printing technology, and all the guidelines present now are for conventional manufacturing. We don't have any guidelines for 3D printing. So in coming future might be some guidelines will be available so that we can judge our product. It is suitable for market or not.
Basic guideline that is not going to change, that generic product has to meet the innovator. B should be passed. And hardly there is any room to play with the excipient. We can play with the 50 or 100 mg max. Okay. So this is the reality actually, I'm telling you.
Yes.
And at the same time, pharma industry is very price sensitive now. In Europe market, if you see that there is tender business. US market is highly competitive. India market is a cutthroat competition. So this is a good new topic you have taken that will of course add value to the existing knowledge, but please keep these points also in mind. This is my suggestion. Sir. Thank you.
Yes, thank you, sir.
Yeah, Gautam here. So it's good. You have taken something different topic, mostly nanoparticles and other synthesis, etc. So it's a good initiation that you are thinking about the customized medicine. Okay, but at the same time, I'm completely agree with Mr. Arun. He is industrialist and he knows about what are the challenges in industries with respect to generic medicines. Okay, so here I just want to know, do you know any product which is already available in the market as based on the 3D printing?
Sir, yes, it is Pretum tablet and it is in the market and it is FDA approved also.
Yeah, so what are the guidelines? You mentioned that guideline is not there. How that product came into the market? So you need to understand what they have characterized and what was the objective of that product, so that you can take certain extract from those information and you can also utilize in your presentation also or in your work also.
Yes.
Okay. So questions are valid. Only you need to find out in what way objective of your formulation. Is it your formulation for the generic product development or for the customized or for the new product development?
It is customized one, sir, new product.
Your answer should be in that way, okay, whether the company will go for the generic product or for the newer product, etc. Okay, for the especially where the dose titration or dose reduction, etc. required based on suppose the cancer patient, etc. is there, where the formulation, like where the clinics can start their own product with that. Okay, in that way it is very useful for that, or future, like as one product is already in the market. So that way you should identify the gap and the rationale for your product.
Yes, sir. Sure, sir.
Right. So those things overall it is good work. So what the existing that market available existing product, it is based on which technique?
So it is binder jet, I think, sir.
Just
Not
check it,
confirmed.
because that also is gap is there, like what they have done and how you are changing the laser sintered. Okay, so how you are making the difference from the existing technology to the new technology.
Yes, sir.
Okay, so that's all from my side. Yeah, thank you. It's good presentation.
And
Thank you, sir.
yeah, Arun here again. To add value to your whatever you have done that is innovative, you can say that this technology can help the innovator companies. Innovator companies means the companies who are working in the phase 2 or phase 3 molecules right now. That molecule may come in the market in next 5 years. So you can sell this concept to them. Then by incorporation of this technology into their innovative product, they can have the product which is difficult to copy. It is like that.
Okay, sir.
Thank you.
Yeah, I totally agree with Mishra sir as well as Gautam sir. A good thought, good concept you're developing on technology. Just I would like to know that what you have done is you have so far in your work that you presented, you have talked about a model dye, right? What I understand.
Yes, sir. Yes, ma'am. IR absorbing dye. It is a laser absorbing dye.
But have you incorporated a drug into that?
No, ma'am. Actually, it is in future perspective, because up till now my work is methodology based. Means this method we are coming with the novel method, and in the next step, once confirming that this method is suitable or feasible for pharmaceutical grade excipients and polymers also, our next step is to incorporate drug in that.
So do you look in for any specific category of drug molecule which will suit for this particular process? Because I understand that you are subjecting the molecule, the manufacturing method was, I mean, you had subjected the mass blend or when you were printing at around 150 degrees Celsius. Correct me if I am wrong.
Yes.
So
yes,
how would that be applicable for molecule which is, I mean, prone to temperature?
No, ma'am. We can change the polymer. This is based on the polymer properties. This polymer we have used. So for this glass transition temperature and melting temperature are high. So the temperature required for its fusion and partial melting is high. We can use some polymers which are having low melting point and low glass transition temperature, so that at that temperature our drug will be stable.
Just one quick question from my side. Is it industry scalable? And if so, would the formulation made out of this technology would be cost effective and at par with existing manufacturing methods? What is your say on that?
Yes, ma'am. It is industry scalable because it is suitable for scale up. This is powder bed fusion technology. Powder we can, as it is used, and we can manufacture in a bulk. And
so do you think that it will be cost effective?
Yes, ma'am, because we are reducing the labor cost, we are reducing the excipient cost, and everything we are reducing. Time also we are reducing. So I think it will be a cost effective. We are in preliminary stages only. This cannot be confirmed, but still up till now I can give confirmation that it will be reduced cost.
What was your processing time, total processing time to get the tablet?
It will be in minutes, ma'am, because we are synthesized in large. It is dependent upon process parameters. We cannot specify that. It will be depending upon process parameters and material we have used.
But roughly, you will finish this process in how much time, based on your experience?
For complete process, one.
Okay. And just have a look, because your disintegration time was quite high, huh? If you're saying for IR tablet. So yeah, that's it from my side. Thank you.
This is my third intervention. Excuse me. You can say that while starting your presentation that we are
making some innovative technology that, that this is a kind of platform technology that can revolutionize the pharma industry in down the line 5-10 years, something like that. And then you can give the presentation that will, add value to that. Thank you.
Yes, yes, completely agree. Yeah, you can take the suggestion very valid
Yeah.
for your future presentation. Then
Totally agree with sir. Yeah.
thank you, Arun sir and Sujata ma'am.
Thank you, sir. Thank you, ma'am.
Thank you, O PhD 10. The next presentation is by O PhD 11.
Yes, ma'am. I'm sharing.
Okay.
Ma'am, am I audible and
Yeah,
visible?
you are visible and audible.
Ma'am, is my slide is visible?
No, your slides are not visible.
Okay. Now, ma'am. Hello.
Yes.
Is it visible?
Yes, it is visible.
Okay. Should I start?
Yes, yeah. You can start. You have 10 minutes.
Okay, ma'am. Okay. Good afternoon, everyone. Today, I am going to present on the topic exploring the potential of adenosine conjugated PLJ nanoparticles for the treatment of triple negative breast cancer. Starting with introduction, first of all, it is difficult to treat the triple negative breast cancer due to some hurdles. As we know, the triple negative breast cancer do not possess the estrogen receptor, progesterone receptor, and HER2 receptor. That's why it is difficult to treat triple negative breast cancer in comparison to normal breast cancer. Since there is the various drugs available to treat Tnbc, one of them is the paclitaxel, and various marketed formulation is also there to treat Tnbc. The one formulation is Abraxane, and the other one is the Genexol. But the major drawbacks related with this marketed formulation is this formulation contains non-ionic surfactant cremophore-EL and ethanol in the ratio of 50-50, which cause the hypersensitivity, neurotoxicity, and nephrotoxicity, as well as cremophore-EL leach out in the IV set that is used for the administration. To overcome such hurdles, we plan to prepare adenosine conjugated pegylated PLJ nanoparticles. The adenosine conjugated pegylated PLJ nanoparticles was prepared using the copolymers that contains the PLJ and the PEG copolymers that was prepared using the ADC-NHS reaction. And the further adenosine was further conjugated with the PEG PLJ copolymer with the same ADC-NHS reaction, which lead to the formation of the carbonidide using the carbonidide reaction. And then PTX drug was loaded into it. And the prepared nanoparticles are prepared using the solvent emulsion evaporation method. And it was characterized for the size, PDI, zeta potential, and percentage drug entrapment efficiency. And it was found that the size was found around 135 nanometer, and percentage entrapment efficiency of the paclitaxel was 79%. And same was also seen in the same analysis and the TEM analysis. The size was also found to be in good coordination with the size obtained from the zeta sizer. And the further, the drug release was performed at the two pH, the pH 7.4 and pH 5.5, that is equivalent to the biological pH and the tumor microenvironment. As shown in the figure, the drug release in the pH 7.4 was found to be less. At the end of the 25 days, the drug release in the case of the pH 7.4 was found to be around 54%. While the drug release in the case of the pH 5.5, that is the tumor microenvironment, it was found to be around 74%. And further, the various in vitro biological characterization was performed. The one of these is the qualitative cellular uptake. Qualitative cellular uptake was performed on the MDMB 231 cell line and 41 cell line. As shown in the figure, as the green fluorescence indicate the enhanced uptake, cellular uptake of the formulation which contains the combined 6. As shown in the D and the E, the enhanced green channels, green fluorescent in the both cell line, it indicates the enhanced cellular internalization of combined 6 loaded formulation. That is the adenosine pegylated PLJ nanoparticles. And the further it was confirmed using the dipyridamole, that is the adenosine receptor blocker. When the receptors was blocked during the dipyridamole, the cell uptake was significantly reduced. And this qualitative uptake was further strengthened with the quantitative cellular uptake, and the same result was found in the quantitative cellular uptake also in the both 41 and MDMB cell line. In this case also, when the cells were pre-treated with the dipyridamole, that is adenosine receptor blocker, a significant reduction in the cell formulation uptake was observed. Similarly, to evaluate the cytotoxicity of the prepared nanoparticles containing drugs, MTT was performed using the MDMB231 cell line and the 41 cell line. And when the drug was treated with the PTX-loaded adenosine conjugated pegylated PLJ nanoparticles, the IC50 value was found to be 3.42, which shows the 3.77-fold reduction in IC50 value in comparison to free PTX in the case of MDMB231 cell line. And similarly, in the case of 41 cell lines, the IC50 value was found to be 3.16, and it shows the 3.51-fold reduction in the IC50 value in comparison to free PTX. And further, the apoptosis study was performed. Apoptosis study was performed using the Anexin V Site 3.1 apoptosis kit. In this kit, the green channel shows the viable cell, red channel shows the apoptotic cell, and as shown in the figure, and the D and the E, that is the formulation. E, that is contains the PTX loaded adenosine conjugated pegylated PLJ nanoparticles. It shows the enhanced red fluorescence, which indicate the cells are more apoptized in comparison to free PTX and other non-conjugated PLJ nanoparticles in the both cell line, that is the MDMB and 41 cell line. It indicates the efficacy of the prepared PTX loaded nanoparticles was found to be more efficient in comparison to free PTX and other. Now, apoptosis index was also calculated. Apoptosis index was calculated using the fluorescent of the red divided by fluorescent intensity of the green. And the apoptosis index in the case of MDMB 231 cell line was found to be 0.93, while in the case of 41 cell line, it was found to be 1.18. And in vivo biological characterization, that is the pharmacokinetic, was performed. Pharmacokinetic was performed using the female HD rat, and it was compared with the marketed formulation in Taxol as well as Nanoxol. When pharmacokinetics parameters such as AUC, T-half, and MRT was calculated, it was found that the prepared formulation shows the better pharmacokinetic parameters in the term of AUC, T-half, and MRT in comparison to marketed formulation in Taxol, Nanoxol, and non-conjugated PLJ nanoparticles. Further, PD study was also performed using the Swiss albino mice female race. In this, the tumor reduction in the tumor volume and the reduction in the percentage tumor burden was calculated. As shown in the figure, as in the case of control, the tumor volume was found to be significantly increased, while in the case of the treated group, there was the significant reduction in the tumor volume was observed in the protocol of the 21 days study. And similarly, the percentage tumor burden for the control was found to be around 8%, while in the case of the treated animal group, which was treated with PTX loaded adenosine conjugated pegylated PLJ nanoparticle, it was found to be 0.3% only. Further, to assess the biological distribution of the formulation, this formulation was loaded with the freecomidine, and it was injected intravenously into the mice, and different vital organs were isolated, and these were observed under the CLSM. As shown in the first case, when the freecomidine is injected, it shows the maximum distribution in the vital organs. That might be the toxic. When it is incorporated into the adenosine conjugated pegylated PLJ nanoparticles, it shows the maximum accumulation in the tumor area and negligible accumulation in the vital organs, which show the prepared formulation will be less toxic in comparison to free PTX and non-conjugated PLJ nanoparticles. And similarly, in the same way, the toxicity study was also performed. The hemolytic toxicity study was performed. In the hemolytic toxicity study, the oxidized hemoglobin absorbance was observed, and it shows that the prepared nanoparticles shows the very less percentage hemolysis in comparison to free PTX and marketed formulation in Taxol. And it was further strengthened using the same image of the RBC. As shown in the image of the RBC, the shape of the control RBC was found to be similar to the treated one. Similarly, toxicity.
PhD 11, 2 minutes left.
Okay, ma'am. Right. Okay. Similarly, the toxicity study was also performed for the different biomarkers such as AST, that is the aspartate transaminase, alanine transaminase, blood urea nitrogen, and creatinine. This study was performed using the kit, the Accurex kit, and their level in the blood serum was calculated. And it was found that when the animals was treated with the prepared nanoparticles, the different biomarkers such as AST, ALT, blood urea nitrogen, and creatinine was found to be somewhat similar to the vehicle, which was given PBS as a vehicle. These biomarkers was found to be similar to the DAT only, which indicate the prepared nano formulation is non-toxic and it is safe to use. Finally, I'm concluding like adenosine receptors that present on the cell surface is upregulated in the various cancer, and it is used for the targeting of the PTX loaded PLJ nanoparticles. And the prepared nanoparticles exhibited the superior in vitro and in vivo efficacy in respect to pharmacokinetic, biodistribution, and safety profile. And I can also say that this can be used as a prototype for the delivery of difficult to deliver hydrophobic drugs that can be used in the further future. These are the references that I have gone through. Thank you.
Now the session is open for Q&A.
Gautam sir.
Dr. Gautam.
Yes, sir. Yeah. So it's good work you have compiled, and only challenge is what like toxicity associated with your particles. Okay. So have you compared with some any other grass limit or any other standard materials?
Sir, we have performed toxicity in 3 PTX along with the various nanoparticles. In the 3 PTX, it is the more toxic. We can take PLGA as a polymer, and we performed the toxicity for that. But it is not shown here, but no toxicity was observed for that polymer also. As in the case of nanoformulation, polymer is also there. But if there will be the toxicity, then it will definitely show the toxicities.
So how will you contribute like when you compare with, just I want to know your thought, when you compare with the polymer and you compare with the formulation in both the cytotoxicity, okay?
Yes, sir.
Which is contributing in your cytotoxicity?
Sir, we perform the free drug as well as the free drug containing nanoformulation. So what we observed that the formulation containing PTX was more toxic. But we also performed the free nanoparticles, but that was not that much toxic, and we forgot to add that result here also. But what we observed, that was not that much toxic.
Okay. So another question is like about the PLGA. Okay. So
yes,
what grade, like what is the composition of PLGA and what ratio of P and GA?
Yes, sir. Yes, sir. So PLJ in the ratio of the 50-50 was used.
50-50. So what is the differentiate you feel like when you use 30-70 or any other ratio and compare to 50-50?
Yes, sir. So we will change the ratio like to 70-30 or 30-70. Then the drug loading or the drug entrapment efficiency will be significantly hampered. When the hypophobicity will increase, then its stability and other parameters will get affected. When we'll decrease like hydrophobic parts of the PLGA, then drug loading might hamper.
Yeah, that is the you are saying the possible regions. But have you checked any at the preliminary level in other ratio?
No, sir, we have not checked in other ratio, but we have optimized for different parameters such as regarding the stabilizers, what should be the stabilizer, what should be the amplitude, and what should be the percentage of the this stabilizer and these things.
Fine. Okay. That's all from my side. Thank you.
Okay.
Mishra sir, Dr. Mishra.
Arun here. Yeah. So we are talking so much about toxicity and all these things. To you and the other, PhD aspirants, I would request that please go through some details of the nitroso impurities, because why pharmaceutical industry is suffering from these nitroso impurities. So at least the our PhD scholars should have some idea about the nitroso also. Okay.
Sure,
So
sure.
that's all from my side. Thank you.
In fact, that would be applicable. I mean, very well said from Mishra sir, because that would be applicable for all the presenters who have presented earlier. Just tap on whether you can do something with respect to nitroso impurities. Because that's really killing pharma industry these days. Couple of products that have been approved, they have to be withdrawn because of this grave issue that is happening. Two questions from my side. Yes, sir.
If somebody does something innovative with respect to nitroso, pharma industry would take that person or institute in hand.
Yeah.
We are
because
badly.
The industry is really getting badly affected with this, and there's a dire need, to quantify, characterize that impurity, qualify it, and the way how we can evade this problem, that is the need of the hour. Two quick questions to you, PhD 11. First, let me congratulate you for the work that you have done with respect to completeness of the work. It's really good. What I would like to ask you is, you have mentioned that your formulation would be effective for triple negative breast cancer. So yes, probably I missed that point. Any specific study that you showed to endorse that your formulation is effective for TNBC?
Yes, ma'am. As MDMB 231 cell line and the 41 cell line, this contains the overexpressed adenosine receptor. And in the case of MDMB 231 cell line, the adenosine receptor A2B is overexpressed, while in the case of 41 cell lines, the adenosine receptor A2, A, and A3. There are the four types of the adenosine receptors, in which A2A and A3 are overexpressed in the 41 cell lines, and A2B are overexpressed in the MDMB 231 cell line. So we performed this cytotoxic MTT study in these two cell lines to demonstrate that is it efficacious or not. So we got the good result in these two overexpressed cell lines.
What is your proposed dosing interval? Because taking Gautam sir's question ahead, yes, with respect to grade of PLGA, I understand you used 50-50 ratio.
Yes, ma'am.
Yeah. So yes, what was your dosing interval? What is if the formulation is there?
In our protocol, the dosing interval is after every 5 days, ma'am.
After every 5 days.
Yes, ma'am.
So in your release profile, how long have you done your release studies?
Ma'am, we have performed release study till 25 days, ma'am.
25 days.
Yes.
Sorry, I missed the point.
Yes, ma'am. 25 days, ma'am.
25 days. Okay.
Yes.
And you what if you change to 75-25 ratio? What profile any?
Ma'am, when we will change to 75 to 25 ratio, the sustained release of the drug will be less. That drug will be released earlier, and their sustainability will be reduced.
Okay. Okay. Thank you.
Thank you, ma'am.
I now request our judges to provide valuable feedback to our participants.
Yeah, Dr. Mishra.
Yeah, so regarding you are asking about the OR.
So just a small feedback.
Yeah, the feedback is that, Three common points like to suggest to this young generation. First of all, what work we are doing, as the panel members have suggested, we should have the clear-cut scheme of work. Okay, this is what we want to do. This is what we want to achieve. Number two, in the presentation, at least there should be some initial second or third slide, which can show that the candidate has done so much work. Now they are presenting a part of their work in this presentation. So that will give an idea to the judges that this work we have candidates have already done. So we will not ask the repeated questions. Third point is that when working on the some PhD project or research project, we should know what is the competition. This competition scenario is missing.
Thank you, sir. So, I would like the judges to receive this small token of appreciation. And thank you so much, Dr. Gautam Singhvi, sir. Thank you, Dr. Arun Kumar, sir.
Thank you.
And thank you, Dr. Sujata Savarkar, ma'am.
Thank you so much, the organizers, for inviting us. And we must congratulate all the presenters. I think Dr. Gautam will share his thoughts definitely.
Yeah.
But I must say, with respect to completeness of the work, it was a good learning experience for me as well. A lot of knowledge sharing and thought-provoking work. So you really churned our interest. So congratulations to all the presenters and all the best for your future endeavors.
Yes.
I would like to add a few words, and I would like to thank all three judges, Dr. Sanghvi, Dr. Arun Kumar Mishra, and Dr. Sujata. Definitely, because I have been listening to all the students' presentation as well as judges' comment. It was indeed a learning experience, and I made sure I took a quick round of college and I made sure that all the current MPharm students as well as PhD students in the campus, they are actively listening to it and learning, because it was indeed a learning experience. Lot of good work was presented, I must say. The quality data, quality research protocols were presented. So I must congratulate each and everyone who presented into this segment. And I must appreciate the inputs of all the three judges. So Dr. Mishra gave inputs from industry perspective, Dr. Singhvi gave inputs from research perspective, as well as Dr. Sujata Madam gave the perspective from research point of view. And it was indeed learning for students also, as well as the presenter. So I wish all the best to all the presenter, and I express on behalf of SVKM, express my thanks to all the three judges and the students, those who have participated across India, and the students who have completely monitoring technically and coordinating the entire activity. So thank you very much, and we appreciate your time and efforts.
Thank you so much. Quick few words from Dr. Gautam Singhvi.
Yeah,
First of all, thank you for joining us.
Yeah, thank you, madam, and thank you, Munira ma'am, and Sujata ma'am, and Dr. Arun Kumar. So this is really a good platform, and using this online as well as plus offline, because morning in the inauguration I was there through online, but it was completely held through offline. Okay, it was good, and I appreciate such events where the students are getting platform. Before passing out, they can improve at every step, they can improve their expression, their presentation skills, their rational thought process, because these events only, they will sensitize them how to think, how to present, how to explain, how to compile the data. So that is really good work. It is doing by SVKM's Dr. Bhanuben, Nanavati College of Pharmacy. And thank you, Munira ma'am, for involving me. and definitely I'm like insufficient or maybe not complete without the inputs of Dr. Sujata and Arun sir. And that all we help. Our intention is to help our students, these new young minds, to come up with a good thought process, express their ideas, present well, explore more in the scientific community. Thank you all. Thank you very much for inviting me.
Thank you so much.
Thank you.
Thank you, Principal Ma'am. And thank you to all the judges for being such an essential part of the event and providing our participants with such critical feedbacks, which will pave the way for the next amazing research work. Last but not the least, I would also like to thank to all the participants for putting forward so much efforts and brilliant work. We hope to see more such works in the coming future. Thank you to everyone, and feedback form link for today's session will be posted in the chat box at the end of the session, and so everyone is requested to fill the feedback form by today only. Certificates will be received for those only who have submitted the feedback form. Now, the next session of the oral presentation competition for the PG category would be continued by my co-host, Vaishnavi. Thank you, everyone.
Thank you, and thank you, Vaishnavi, for conducting and coordinating this event very fruitfully. Yeah.
Yeah, thank you, sir. Thank you.
Good afternoon, everyone. I, Vaishnavi Golakar, would like to welcome everyone to the second session of the third Student Research Congress organized by SVKM's Dr. Banuben Nanavati College of Pharmacy, co-hosted by University of Mumbai, Industry Partner, ACG Worldwide, and Cytec Center. As it is rightly said, the only way of discovering the limits of the possible is to venture a little way past them into the impossible. So now we will be having oral presentation competition for the PG category. Let us begin the session with the introduction of the judges for the oral presentation competition PG category. Our first judge for today is Dr. Hema Naya ma'am. Ma'am has an experience of 15 years at Bombay College of Pharmacy, Mumbai, followed by a move to Hyderabad and currently associated with Sri Venkateshwara College of Pharmacy as Associate Professor. She has pursued her B.Pharm and her M.Pharm from University of Mumbai and her PhD from the SNDT University of Mumbai. Ma'am also have a diploma in patent law from Nalsar Law University. She has guided a total number of 13 M.Pharm students as well as 2 PhD students. Ma'am have 3 national research papers, 14 international research papers, including 2 of which have been awarded. Along with this, ma'am also has many number of research and review publications. Ma'am has received research grants as Chief Investigator, totaling more than 50 lakhs from AICTE, UGC, University of Mumbai, ICMR, IGC, UGC, RGNF, as co-investigator, totaling more than 75 lakhs from AICTE, BRNS, DBT. She also have the membership of professional bodies like IPA and APTI. Ma'am is also the member of editorial board of Indian Journal of Pharmaceutical Sciences. Also, she has been reviewer for several national and international journals. Our second judge for today is Dr. Manju Mishra ma'am. Ma'am is currently associated with Graduate School of Pharmacy, Gujarat Technical University as an associate professor. She has pursued her B.Pharm and M.Pharm degree from Nagpur University with specialization in pharmaceutics. Ma'am has pursued PhD from BIT Mesra, Ranchi, and her research project was working on thermal analysis and its application on drug excipient compatibility screening. She has total 15 years of experience post PhD. Also, she has the experience in various research, formulation development, and development unit of pharmaceutical industries like Macleod Pharmaceuticals Mumbai, Piramal Pharmaceuticals Development Service Private Limited, Ahmedabad, Cadila Pharmaceuticals Private Limited, Ahmedabad, wherein she worked in generic formulation development for regulated market. Post her industrial stint, she joined NIPER Ahmedabad, an institute of national importance in the area of pharmaceutical research, under the aegis of Ministry of Chemical and Fertilizers, Government of India, as assistant professor in the Department of Pharmaceutics, where she worked for 10 years. At NIPER, ma'am was instrumental in setting up entire pharmaceutics department and played key role in several academic and administrative responsibilities, including subject coordinator for pharmaceutics, NIPER academic course coordinator, placement coordinator, coordinator for industry academic linkage, chairman of NIPER JEE 2018, and several others, apart from teaching and mentoring MS and PhD students. Our third judge for the day is Dr. Tabassum Khan ma'am. Ma'am is currently affiliated to SVKM's Dr. Bhanuben Nanavati College of Pharmacy, Mumbai, as professor in HOD, Department of Pharmaceutical Chemistry and Quality Assurance. Ma'am has pursued her PhD in Tech in Pharmaceutical Chemistry as a Senior Research Fellow at CU Shah College of Pharmacy. She was working as a scientist in research and development at Procter & Gamble India Limited, Mumbai. Her teaching experience is 23 years and industry experience of 4 years. Ma'am has received research funding of 24.6 million INR from agencies such as DST FIST, Ayush CCRUM, PRNS DAE, DST SERV, RGSTC, University of Mumbai. She has published 70 papers in international and national journals with total impact factor of more than 120. Talking about her academia career, currently she is guiding 5 PhD students and has guided 27 MPharm students. Ma'am has been invited as a reviewer for reputed international and national journals. She has also been a referee for international research projects in Poland, Serbia, and Canada. She has received Shastri Mobility Program SMP Fellowship Award for the year 2017-18 from Shastri Indo-Canadian Institute. With this, we would like to welcome our judges with a bouquet. A hearty welcome to all the judges. I would now like to draw the attention of the participants to the instructions. Rename yourself as per the allotted course, if not done already. Participants cannot reveal their name or any affiliation. Each presenting author will be given a time of 10 minutes for the presentation. Follow the time limit and kindly do not exceed. Only the presenting author will be allowed to answer in the Q&A session. Each participant is requested to stay alert for the roll call. With the permission of all the judges, we would like to begin today's session of oral presentation competition, which all the participants have been actively waiting for.
Yeah, you can go ahead.
I hope all the judges have received their scorecards.
Yeah.
Okay, ma'am.
Yes.
Judges, there has been a slight change. So there were total 7 participants selected, but 2 of them have cancelled due to the health issues, and 1 of them we are not able to connect. So today we will be having only 4 presentations. So now I would like to call our first PG presentation of OPG1. OPG1.
Hello.
Yes, you're audible.
Yes.
Can you switch on your camera?
Yes.
OPG.
Yes. Am I visible?
Yes, you're visible. Please share your PPT.
Yes, is my PPT visible?
Yes, it is visible.
Yes, so I'll start. A very warm good afternoon to all judges and participants. Today title for my today's presentation is Amorphous Solid Dispersion of Mesalamine in a Novel Carrier of Mesoporous Silica, an attempt to ameliorate the incidence of ulcerative colitis. So the table of contents are introduction, aim and objectives, materials, formulation and method of preparation, method, results and discussion, and references. So coming to the introduction, in this project we have used mesalamine as a drug to form a solid dispersion using a mesoporous silica. So a mesalamine is a BCS4 drug used in a first-line treatment for anti-inflammatory effect in colon-related disease like ulcerative colitis and Crohn's disease. It appears to act locally on colonic mucosa and reduce inflammation. Hence, the localization of the drug at the site is desired for healing. For effective localization, the primary need is to improving and maintaining the solubility of the for the desired period of time. Solid dispersion is a dispersion of drug in a carrier matrix, and adapted method because it is easy to produce and reproducible. Uh, in the present study for amorphization process, novel carriers, different grades of mesoporous silica were being applied. Mesoporous silica is the solid framework of SiO bond with a porous structure and large surface area. It has a unique feature of offer high drug loading capacity and it is FDA approved. Biodegradable, tunable pore size, more stable, high drug loading capacity, and large surface area are few of the benefits of mesoporous silica. Hence, the present study aims to form delayed release solid dispersion of mesalamine in a mesoporous silica to improve the drug release and localized effect of the drug in a ulcerative colon. So the objective of my study is to prepare an amorphous form of mesalamine using mesoporous silica to observe the effect on the improvement of solubility and thereby dissolution of the drug. To estimate the compression characteristics, solid state, surface behavior and local effect on the ulcerative colon in animal through histopathology study. Coming to material and method, different material used are drug mesalamine, three different grades of mesoporous silica like Siloid 244 FP, Siloid XGP, Nucilin US32, and coating material Utragic E30D5. Nine different formulations were prepared by taking different ratio of drug to form a monolayer, double layer, and triple layer. The ratio was calculated by using Dennings and Taylor equation. In the right bottom, we can see the general method of preparation of solid dispersion via solvent evaporation method. In this, the drug and polymer was incorporated in ethanol to form a slurry, and then the slurry was kept on a mechanical shaker for 24 hours. And to evaporate the solvent, it was kept on rota evaporator to get a solid dispersion. These are the few evaluation which was carried out for the solid dispersion. Method, results and discussion. Compatibility study was carried out by FTIR using instrument Bruker model alpha 2 at a range of 4000 to 400 centimeter inverse. Here FTIR spectrum of drug and different formulations are given and all the characteristic bands of pure drug were preserved and indicate compatibility of drug with different grades of mesoporous silica. Estimation of drug loading in mesoporous silica. It was performed by taking the slurry and then centrifuge at 10,000 rpm for 15 minutes. Then the superlated liquid obtained was diluted in PBS 6.8 suitably and was analyzed at 330 nanometer and percentage drug loading was calculated. The drug loading was given in a graphical manner. The white difference in the drug loading is an indication of the physisorption on the nanopores of silica at different layer. The maximum loading was seen in the triple layer formulation. Estimation of flow property and compressibility, bulk density, tap density, Hausner's ratio, Carr index, and angle of repose were performed. All the formulations showed a good flow property and compression characteristics. Solubility study. In this, a maximum dose equivalent of solid dispersion was dissolved in phosphate buffer solution pH 6.8 and kept on a mechanical shaker for 24 hours. Then it was suitably diluted in phosphate buffer solution 6.8 and analyzed at 330 nanometers. There was significantly increase in solubility was observed with the carriers Siloid 244 Fp and Siloid XTP. The solubility of the formulation was found to be increased as the layer of the drug loading was increased. Preliminary in vitro drug release study, it was performed by filling the solid dispersion in a capsule and then the dissolution was performed in buffer PBS 6.8. Here you can see the uh, data for the release study of different carrier. All the formulations showed the increased drug release when compared to the pure drug. The maximum release was obtained in the following order. Siloid was Siloid 244 Fp greater than Siloid XTP, greater than Mucilin US32 and greater than pure drug. Then statistical interpretation was carried out by taking key parameters like drug loading, solubility, drug release and compressibility, and the p-value obtained was much less than 0.05. Then further Dunnett comparison test was carried out between formulation TSFP, TXGP, TNEU. And Dunnett, it reveals that the TSFP was significantly better than TNEU, but the properties of TSFP and TXDP were comparable to each other. Hence, for further study, TSFP and TXDP were chosen. Scanning electron microscopy was performed to study the surface morphology, and it revealed the crystalline nature of the drug and successful drug loading in the formulation. PXRD was performed using Bruker's D8 Advanced at a range of 4 degree to 60 degree at a rate of 0.002 per minute 2 theta. The intensity obtained for the formulation was much less than pure drug, which indicates the conversion of amorphization of drug and reduction of crystallinity. This might be the reason for improvement of solubility and dissolution. In vitro dissolution of entry-coated capsule. It was performed by forming a coating solution. Then the solid dispersion was filled in a capsule and then subjected to spraying by spray coating method. And then it was carried for dissolution using USP procedure. In USP procedure, it was carried out by 3 different buffers. First buffer was acid stage 1. In this, HCl of pH 1.2 was taken 500 ml for 2 hours at 100 rpm and 37 degrees Celsius. Then the buffer was changed to 900 ml of phosphate buffer for 100 rpm for 1 hour at 37 degrees Celsius. Then additional 50 ml of NaOH was introduced to increase the pH. As the result, we can see here in the graphical manner, first initial 120 minutes there was no release of drug. This indicates the integrity of the entry coating. Then a shoot release was seen in the pH 6 and a steady in 7.2.
2 minutes are remaining.
Okay. BT study was carried out to see the specific surface area, and the specific surface area was found to be increased remarkably in the formulation TSFP compared to TXDP. The isotherm exhibits a type of 4 curve which indicative of physisorption. The increase in the specific surface area is attributed to higher solubility and drug release. Histopathology study of TSFP, it was carried out using albino rats of 200 to 250 grams. In these 4 different groups were formed containing 6 rats in each group. The group A was served as control and group B, C and D was induced with ulcer using 5% acetic acid in a saline solution. Then group C was given a treatment with the pure drug and D were given our formulation and was analyzed with the scoring at 6 hours and 24 hours. The formulation show a potential decrease in the ulcer over a long period compared to the pure drug. Hence, the localized healing action of formulation TSFP on the ulcerative colon was enhanced. Then the stability study was carried out as per ICH Q1A guidelines. For the formulation was kept for 90 days at 25 degrees Celsius plus minus 2 degree and 60% plus minus 5% relative humidity, and the dissolution was carried out by USP procedure. The result indicates there was no significant change in the dissolution data at the end of 3 months storage period. Hence, the stability of the product was established. Coming to the conclusion, summary and conclusion, For the solid dispersion, the drug loading was enhanced, improvement in the flow property was seen, improvement in the solubility in the triple layer was seen, and resolution when compared to the pure drug was increased. Amorphization was successful in PXRD and SEM. In statistical, we
Your
revealed.
time is up. The
And
session
last,
is
I
now
would like to
open.
conclude that the delayed release solid dispersion of mesalamine in a mesoporous silica could ameliorate the incidence of ulcerative colitis. These are some of the references, and thank you. That's all from
Thank you,
my side.
PG1. The session is now open for the Q&A.
Hema and Manju, please go ahead.
Yeah. Can I
I'll be the
go
last
first?
person to ask.
Yeah, okay. Okay. So couple of first thing is that was a very elaborate amount of work for an MPharm presentation. So congratulations. Good theme
Thank you,
also. Just a few points I need you to clarify. Right. So
Yes,
first thing is you have enclosed your material into the capsule and enteric coated.
Yes, yes.
How is this one step of enteric coating going to ensure that your drug reaches the colon, because it's a very locally acting drug? It needs to act in the colon, right?
Yes, yes. Yes,
So-
ma'am. So, yes, so in release study where we have done with enteric coating capsule, we can finally see that in initial 120 minutes where there was acidic pH, which means
Yeah, so
stomach.
that is okay, but at the
Yes.
end of 120 minutes, the whole thing is coming out.
Yes, the release, yeah, we can see a shoot release. Yes.
Yeah, it's a shoot release. So how after that it is several more hours till it reaches the colon?
Yes, ma'am.
So I was just wondering about the rationale of maybe not coating it further, or adding a coat within your enteric coat so as to ensure a slightly more delayed release. And okay, I'm not aware of the absorption profile of mesalamine and all that, but it is if you increase its solubility in the part of the intestine which is very good at absorption. Okay.
Yes.
That might not be the right approach, because the drug could get absorbed, whereas it is a locally acting agent
Yes,
required to be delivered to the colon.
Yes, it shows localized action.
Yeah, so these are a couple of things that I found. Another thing is you claim it's a triple layer absorption.
Yes.
How did you come to that conclusion? Did you present that here just now?
Sorry, ma'am.
You said that it was a triple layer absorption, layer, which was where the loading was the highest.
Yes, yes.
How did you conclude that it's a triple layered product?
Ma'am, we have done experiments with 3 different layers. So in first layer, second layer, and triple layer, and then we have statistically seen then that the third layer was improved and it was showing better results. So for further study, we have carried triple layers only.
Okay, that's okay. In your preparation, you have mentioned a polymer. That's actually what you mean by silica, right? Silica is not a polymer, but you have
No.
your
Udragit.
picture shows Udragit.
Yes, it is the polymer, ma'am.
No, for the preparation of the particles. Not for
No,
coating.
that is silica. No, silica we have used.
Silica, but you have labeled it as a polymer in your graphical representation.
Okay, I'll look into that.
Yeah. Okay. That's it from me. All the best.
Thank you, ma'am.
Yes. Yeah, contestant 1. You have used 3 different grades of silica: xyloid, and nucellin. What is the difference in the 3 different grades?
Ma'am, these 3 are very minorly different because it belongs to same category of mesoporous. There are 3 different, they mainly differ in their structure. So like MCM4
surface area, particle size.
Sorry?
They differ in structure, surface area,
Yes.
or particle size.
No, these are same grades, ma'am. They are of same structure.
Same structure. So they differ in particle size and surface area?
No, it differs very minutely.
So why have you taken 3 grades in that case?
It differs, ma'am. Just to check which one is giving good, better result.
Okay. And the final formulation contains which? Sorry, I missed out that part. The final formulation
Silent.
contains which grade?
Silent
Silent.
244 FP.
What does 344 stand for?
No, ma'am. I don't have idea.
Okay. And again, as Madam has already, Dr. Naya, Madam has already said that the dissolution profile doesn't looks like that it is going to go to colon. If you want it to go to colon for the sake of ulcerative colitis, it is not
Yes.
going there. Probably some trigger has to be there which will facilitate that the release is occurring only in the colonic region and not in the small intestine region.
Okay,
Okay.
I'll look into that, ma'am.
Okay, and what was the method for preparation of solid dispersion? How did you made it?
Solvent evaporation method we used, ma'am.
Okay, and you checked out that there are no traces of solvent at the end. Did you did that?
We carried out, like specifically we have not done.
Okay, no issues. मैडम आप पूछ लीजिए तो बस मैम आप पूछिए मैडम शायद आपने म्यूट नहीं I did it. So I just wanted to know why did you select solid dispersion as your modality of enhancing solubility?
Ma'am, because
any specific reasons?
As we found in literature, it was very easy to produce in lab, and it is very reproducible result, and it is assured for many formulation solubility enhancement. So we choose this method.
And why mesoporous silicon? Why not
It is,
any other technique?
Yes, yes. So it is a very new material carrier. So we thought to experiment with it.
Okay. Any limitations of both solid dispersions and mesoporous silica?
Yes, there are limitations after storing. The solid dispersion may convert into crystalline. It is a disadvantage. As we can see, there are different generations of carriers we have used. So we have take care of that by storing, like suitably storing that, and that's why we carried out the stability study at the end, just to ensure the stability of the product. It has converted into crystalline or not.
Okay, last question. Why this drug?
This drug, ma'am, because in colonic ulcer, this drug has been very hot and first line treatment. So I thought to experiment with it.
I don't know.
It sounds like a
model
drug. It sounds like a model drug. No. There has to be a reason. No, other than of the battery of drugs which are there. Why did you do any particular reason for this? Are there no reports of such type for this drug and they are there for others? That is what. There are many papers on solid dispersion of mesalamine. बहुत सारे हैं.
Yes.
Mesoporous के साथ नहीं है.
Mesoporous के साथ नहीं. So that's why we use that carrier. And it is a new carrier, so we thought.
The carrier ki toxicity, you have not told about the toxicity of carrier. You did told about the limitation of the solid dispersion. What is the toxicity of the carrier?
No, we have not done any toxicity study.
नहीं पर आपको पता है literature mentioned what is the toxicity of using mesoporous silica. Are there some concerns
I have not
with that?
Okay, ma'am.
कोई नहीं. Okay. Thank you. Good work, but. Good work.
Thank you, ma'am.
Thank you, judges. Thank you, OPG1. Now I would like to call OPG2 for the presentation. OPG2. Okay, slide change. I'd like to call OPG4. OPG4, are you there?
Hello, am I audible?
Yes.
Yes.
Please share your presentation.
Yes. Shall I start?
Your screen is not visible. Just try to reshare it again.
Yes. Is it visible now?
No, it's not. Should we try to share it for you?
Yeah, if possible. Is it now visible?
No, it's not visible. Okay, we'll share. OPG4, you can start.
Where is my slide? I can't see.
The slide is visible to us.
Okay, okay, ma'am. My topic is Mesil, a novel pharmaceutical medicine for alleviating hyperthyroidism. Next slide, please. The aim of the flow of my presentation will be first aim, then introduction, materials and methods, results and conclusion. The aim of my study is to assess the effectiveness of prepared novel formulation containing silymarin and methimazole in levothyroxine-induced hyperthyroid rats. Next slide, please. Now, hyperthyroidism is a pathological disorder in which excess thyroid hormone is synthesized and secreted by the thyroid gland. The incidence and prevalence of hyperthyroidism is difficult to establish globally. But in India, the prevalence of hyperthyroidism is 1.9%. Now, the methimazole and propylthiouracil, that are thyroid peroxidase inhibitors, are highly prescribed antithyroid drugs, and they have reported to cause hepatotoxicity. An analytical method of silymarin and methimazole was developed, and a novel combination formulation was prepared, and the parameters were evaluated. Now, methimazole is a first-line antithyroid medication for the therapy of hyperthyroidism.
You're not audible.
Hello?
Tabasum, ma'am, she's audible at my end. I don't know about yours.
Yes, ma'am, she is audible.
She's audible to me also.
Achha, she's audible?
Yes.
Oh, okay. Okay, go ahead.
Silymarin being hepatoprotective, it can regulate the thyroid hormones by enhancing the conversion of T4 to T3 in liver and improve thyroid and body functions. Next slide, please. Materials and methods. We used healthy female Vista rats weighing 170 plus or minus 20 grams, and they were used for the experiments after 5 days of acclimatization. The animals were housed in the animal house and under controlled conditions, and the inducing agent was thyroxine T4 at a dose of 0.3 mg per kg for 12 consecutive days except normal control animals. Next slide, please. This is our experimental design. The female Vista rats were randomly allocated into 5 groups, each group containing 6 rats. The first group received water. The second group, that is model control, received 0.3 mg per kg T4
subcutaneously for 12 days, and group 3, that is treatment control 1, that received our prepared formulation after for 15 days after the hyperthyroidism was induced. The group 4, that is the treatment control 2 group, it received only methimazole, and group 5 received only silymarin. Next slide, please. And after the completion of the protocol, the blood samples were collected and serum was separated for estimation of various biochemical parameters like T3 levels, T4, AST, ALT, and bilirubin. And the animals were sacrificed and selective organs like liver, thyroid, and parathyroid tissue were isolated and stored in 10% formalin for histopathology examination. In statistical analysis, the results were expressed as mean, standard error of mean. The statistical analysis was carried out by applying one-way ANOVA followed by the post hoc Dunet's test using GraphPad Prism. Now my results, effects on blood pressure. The T4 treated animals showed significant elevation in blood pressure as compared to normal control, whereas the treatment with formulation methimazole and silymarin displayed decrease in blood pressure as compared to model control animals. This hashtag indicates that there are significant differences from the normal control group, and the asterisk indicates that there are significant differences from the model control group. Next is effect on thyroid function test. The model group was found to have unbalanced T3 and T4 levels as compared to the normal animals. However, after giving the formulation, and methimazole and silymarin, they showed decrease in T3 and T4 level as compared to the model control. Next is effect on liver function test. The thyroxine treated rats displayed rise in serum AST and ALT and bilirubin level as compared to the normal control. Also, methimazole treated rats showed change in levels of AST, ALT, and bilirubin, hence confirming its hepatotoxicity potential. And the animals, those were treated with the formulation and methimazole and silymarin, they exhibited improvement in liver function test by decreasing the levels of AST, ALT, and bilirubin. Next is oxidative stress parameters. Now the animals, those were treated with thyroxine significantly increased the pro-oxidant level MDA as compared to the normal control group. And treatment control animals showed significant decrease in MDA level, that is the oxidative stress marker. Now the model control group showed decrease in enzymatic activity of SOD and catalase as compared to the normal control. And however, the treatment with the formulation methimazole and silymarin has increased antioxidant enzyme activity than that of the model control group. Next is effect on histopathology of organs. First, normal histology of the liver, thyroid, and parathyroid was seen in the normal control animals. Now, marked atrophic changes were noted, like decrease in sinusoidal space due to hepatocyte hyperplasia in the liver. Then decrease in the cross thickness and follicular lining epithelium thickness were detected in thyroid glands. And it was observed in LT4 controlled rats. And the animals that were treated showed a dramatic inhibition in the pathologic changes in their organs. Now, in conclusion, all the treatment groups showed antihypertensive effect as evident by reduction in the blood pressure. The animals that were treated with the formulation and silymarin exhibited improvement in liver function tests by decreasing the levels of AST, ALT, and bilirubin. Now, the oral administration of all three treatments to hyperthyroid rats has recovered serum T3 and T4 levels. Furthermore, they protect against liver injury by combating the oxidative stress as apparent by the levels of antioxidant enzymes and histopathology of the liver. Now, these outcomes suggest that formulation of methimazole and silymarin may help in improvement of hyperthyroidism and liver damage. Therefore, this formulation could be an alternative therapy of hyperthyroidism. Thank you.
The session is now open for Q&A.
Yeah. Do I go?
Yeah, please.
Okay, thank you. Just keep the slide here. There is an error which is actually very big, because you see here in your last sentence, you say these outcomes suggest that formulation will improve hypothyroidism and liver damage. It will improve hyperthyroidism, but it should not improve liver damage, right?
Sorry.
Okay, that was on the lighter side. Now, how did you, your intention of including silymarin was to protect, render a hepatoprotective effect, right?
Yes.
So in what ratio did you combine these two, and what was the rationale behind choosing that ratio?
The ratio was 1:1.
What was the rationale behind combining in that ratio? How do you know that, adding a greater amount of silymarin, say 1:2, would not improve it further? That's what I'm trying to ask you. How did you work out the rationale behind choosing the ratio?
Did you have equal amounts of both methimazole and silymarin?
Yeah, that's what she's saying. Yeah.
The combination was 60 milligram of methimazole and 200 mg of silymarin.
Well, then that's not 1:1, no? On a
It's
weight by weight basis.
1:3.
It's a little more than 1:3. So that's what I was trying to ask you. Whatever the ratio, you said it's 1:1. Whatever the ratio, what was the rationale of combining in those two quantities? So the drug has a dose, right? But silymarin you could have varied. How did you choose 200 mg? Is it the reported dose that you went for? Okay, that's fine. There's another thing. Does adding silymarin also improve the anti-hyperthyroid activity of the drug?
Yes, it improves in peripheral conversion of T4 to T3. So it adds on to that.
Through that mechanism, it also contributes.
Yes.
And you are getting that synergistic effect in your animal work?
Yes.
Okay. Okay, good. Good. All the best.
Thank you.
Yes, just one question. You have got the silymarin, it was extracted by someone else, or you procured it ready-made? I mean, what was the source of this?
Actually, formulation part I'm not aware of. I'm a pharmacology student, so.
Okay.
No, it's fine, no issues. But still, you have administered it to animal, so you must be knowing what exactly is the composition of it. I mean, what is the drug? Methimazole, okay? Methimazole. So methimazole was administered along with this drug. So it was a kind of one after the other, or both
No, it was a prepared tablet. Tablet was prepared.
Acha, you have prepared a tablet of both actives?
Yes. It was a combination tablet.
Okay, and it was administered simultaneously after a crushing as per the body weight of the animal?
Yes, mg per kg.
You have any idea about why the formulation team has actually formulated the two drugs combined together? Was it for you only that combined that particular drugs?
No. It was the project started from formulation and then seeing
you have
the
any
effect on
hyperthyroidism. What was the rationale of the formulation team? Why did they try to combine these two actives?
They wanted to combine because methimazole is known to cause hepatotoxicity. So they try to lessen that side effect, and so they try to combine both of these. And
that's why you mentioned that the ratio was 1:3 of methimazole versus silymarin. The ratio was 1:3. Okay. One more thing, your histological slides, none of them have got some, you just insert appropriate rows, or for that matter, indicators like arrows kind of thing, to indicate what kind of histological features you're trying to discuss, because we sitting here will not be able to kind of find out what is reducing, what is increasing, and like that, okay? So when you are presenting histological slide, please insert arrows at appropriate places that, okay, nucleus expanded, it has vanished or something like that. Okay.
Yes, ma'am. Next time I'll surely implement that.
So I had this question. Was this study approved by the ethics committee?
Yes, it was approved by.
Have you mentioned the approval details in your presentation? I did not see them. That's why I'm asking.
No, I haven't. But
yeah, so do that. That's very important part because it's pharmacology, right?
Yes,
the other thing is, did you have a control which had only the methimazole and silymarin in the absence of a formulation?
Yes, there were 5 groups.
No, the combination alone, not in the formulation.
Means separately.
Just to see the effect of how it does without the formulation. Okay.
Yes, normal control group was there without formulation.
Which group is that received formulation?
First group, normal control group.
No,
that received only water.
No. I'm saying only methimazole 60 mg and silymarin 200 mg. Did you have a group only of those drugs?
Yes. Group 4th and 5th, group 4th and group 5th. Group 4th received only methimazole 5 mg per kg, and group 5 received only silymarin 200 mg per kg.
Okay. So again, this study, this combination is reported. So you've done this because the formulation of that is not reported. Because I just got a paper on the same study, in the same dose, in the same combination. How did you give the tablet? Did you make a suspension or something? How did you administer it orally?
It was crushed and dissolved in the solvent and water, and then it was given orally.
Okay, fine. Thank you.
Okay.
That's all from my side.
Judges, thank you OPHD 4. Now I would like to call OPHD 5 for the presentation.
Hello ma'am, am I audible?
OPHD 5, can you switch on your camera?
Yes ma'am, yes. Yes ma'am.
Okay. Share your presentation.
Yes ma'am. My slides are visible?
Yes, they are visible.
Okay. Yeah. So good afternoon all judges. My topic of presentation is ultra deformable vesicle mediated localized transdermal delivery of docetaxel for breast cancer therapy. So coming to its introduction, breast cancer remains the most widely diagnosed malignancy in the women, and the major treatment is the chemotherapy and in early and advanced breast cancer. But there are some shortcomings. So coming to the purpose of these studies, so docetaxel is the first line antimitotic treatment for the breast cancer, and Receptor is the IV marketed formulation. But there are some problems associated with it, like it has a high systemic toxicity due to its non-specific biodistribution into the various vital organs, and it has a lower exposure in the breast region due to presence of drug diffusion barrier like milk duct. So to overcome these problems, there is a need to deliver this drug through localized transdermal delivery into the breast region. But there are some barrier to this route, like the skin stratum corneum and the physicochemical properties of the drug and its biocompatibility issues with the skin. So we have developed the docetaxel loaded ultra deformable vesicles, and further they are loaded into gel for the ease of application. These vesicles are prepared by cocktail combination of nano-sized vesicles and synergistic combination of permeations enhancers. So coming to its mechanism of action, when DTX loaded UDV when applied to the skin, they firstly alter the stratum corneum structure. After that, there increased deformability, and due to which the enhanced permeation occur. After that, it will reform crossing the skin barrier. So coming to its preparation, and they are prepared by the thin film hydration method, in which the lipid and scope mixture are in the ratio of 8:2 weight by weight. And after that, their film are dried by rotavapor and further rehydrated by PBS 7.4. Later, the probe sonication is done for the size reduction, and it was analyzed for size, PDI, and entrapment efficiency. So the coming to its preparation and optimization, various combination of permeation enhancer like sodium oleate, SLES, and propylene glycol are utilized. And according to the size, PDI, deformability index, and percentage DTX permeated, 64 by 16 to 20% weight by weight show the maximum deformability index, minimum particle size, minimum PDI, and highest amount of percentage DTX permeated. So coming to its drug loading and entrapment efficiency optimization, increasing amount of drug is added into up to 5 to 20%, and at 15% drug added, it show the highest practical drug loading, and after that it showed it was decreasing in the drug loading, and also it was showing the increase in size and PDI. So we have confirmed with that 15% drug loading is utilized for the further evaluation. Then TEM images revealed that the spherical shape of the vesicles. Also to check the elemental analysis, the sodium and sulfur peak in the EDS spectrum, which is electric X-ray dispersive spectroscopy, indicate that there is a formation of uniform vesicle with a phospholipid component. So coming to its preparation and characterization, the UDV DTX is further loaded into a gel in carbopol gel, and it is further characterized for its rheology and spreadability. So in rheology, the drug loaded UDV compared to the other formulation showing the in drug loaded UDV showing the pseudoplastic flow with increasing rate of shear, and also it is showing in decrease in velocity, which confirm that it has a pseudoplastic behavior, which is essential for topical therapy. Also, its spreadability is determined by acrylic plate method, in which the increasing amount of weight is used, and drug loaded HPV show the increased amount of area, which confirm the efficacy, which confirm the ease of application in the topically onto the skin. After that, the release studies are done, in which the dialysis bag of molecular weight cutoff 12 kDs are used, and the release media was pH 7.4 PBS, and it contained the 0.5% 2 in 80 to maintain zinc condition, and the system was maintained at 37 degrees Celsius. And the free DTX and scope mixture was showing the rapid release profile, whereas the drug loaded liposome and UDV showing the sustained release profile. And when the UDV was further loaded into a gel, it was showing the further sustaining the profile up to 76%. And these in vitro release kinetics are further taken into the release kinetic model, in these models the R square value, in which the highest linearity we got in the Higuchi model, and also the N value, which is less than the 0.5, signifies that the UDVs and UDVs gel are showing the diffusion based release of the drug. So coming to its ex vivo permeation studies and deposition studies, these UDV based gel showing the highest permeation in the pig ear skin, and they were showing the highest flux, permeability coefficient, and less lag time and highest enhancement ratio compared to the free DTX scope and liposomal gel. So coming to its skin deposition studies, the DTX loaded UDV gel was showing the highest amount of intradermal accumulation into the visible layer, viable layer, and stratum corneum, whereas a free drug in the gel was showing negligible amount of drug. And coming to its cellular uptake studies, the C6 loaded formulation was used and the cell lines was MDA MB231 human breast cell cancer line, and the C6 loaded UDB was showing the highest fluorescence intensity in the D figure, and compared to its liposomal, scope mixture and free drug. And also its cell viability studies has confirmed also DTX UTV according to IC50 value was showing the lowest value, which confirm its therapeutic efficacy due to the high cellular internalization. So coming to its pharmacokinetic profile, which was done in Sprague-Dowley rat, ultra deformable vesicles was showing the highest Cmax, highest AUC, T-half and mean residence times, which confirm that its therapeutic efficacy compared to its free drug in gel, scope in gel, liposomal gel, and its dispersion. So coming to its efficacy studies, in which the 41 breast cancer induced model was used, and it was compared, UDB gel was compared with the marketed formulation of Taxotere, which is IV, and it was showing the 4-fold reduction compared to the control group, and also it was showing the similar result compared to the Taxotere IV formulation, which can be similarly seen in figure 2 and figure 3. Also in figure 4, it is evident that the UDB gel was showing no weight variation, and in Taxotere it was showing the weight variation profile, which indicate due to its high systemic toxicity. So through this we can conclude that the UDB gel is also safe. So coming to its bioimaging analysis, which is done mainly to check the tumor penetration potential. So we can clearly see that in figure F and G that the UDV gel in dispersion and gel form show the highest fluorescent intensity.
Only 2 minutes are remaining.
Okay, ma'am. And also in figure G, it is confirmed that it was showing the highest intensity, fluorescence intensity, than in dispersion form, because in gel form the adherence was high. And coming to its conclusion, and this prepared UDV gel formulation exhibit the enhanced transdermal flux, ability to penetrate deeper layer of the skin, extended release profile of DTS compared to other formulation was seen. Also, overall results of in vivo and in vitro was done, and it was also compared with the marketed Taxotere formulation, and it confirmed that this formulation has a low systemic toxicity and it possesses the acceptable skin compatibility. These are the references. Thank you.
Thank you, OPG5. The session is now open for Q&A.
Do I go?
Yeah, Hema.
Okay. So OPG, बहुत काम है जी in your at your MPharm level. It's a lot of work. So congratulations and really commendable. Just few technical doubts which come to my mind. Your final product is the ultra deformable liposomes which are embedded in the gel, carbopol gel.
Yes, ma'am.
Right. Now, what makes you think that the drug will not leach out into the gel during storage, especially since you're having surfactant molecules within the liposome, which you have added for the ultra deformable and skin penetration enhancement? Would they not encourage the drug to leach into the carbopol gel during storage?
Ma'am, to confirm this, we have done also the stability studies in room temperature and also in the refrigerated conditions, but I have not included the data here. But the storage stability studies were done according to check the PDI size and entrapment after its storage, to check whether the size is changing or its morphology is changing or PDI. So it was found that there was no significant differences after or before. So yeah.
So you mean to say that even after storage, the drug remains within the liposome and is not leaching out into the cell?
Yes, ma'am. There was no. Yes, ma'am. The significant amount of loss was not there. It surely can deliver the optimum therapeutic dose.
And what is the scope gel, which was one of your controls in the experiment?
Yes, ma'am. The scope was the synergistic permission enhancers, which I included into gel to check whether the effects are occurring due to the permeation enhancers or not.
Acha, so into the carbopol gel you added your things and then you added the drug also.
Yes, ma'am, yes.
What was the volume of your release medium for the release study?
Ma'am, it was 30 ml PBS, 7.4.
Okay. There is an ex vivo study which you have described wherein you have done the deposition studies.
Yes, ma'am, yes.
What was the skin model there?
Ma'am, it was pig ear skin.
Acha, that was also pig ear skin and the diffusion was also across pig ear skin?
Yes, ma'am. Actually, the skin which were used in skin permeation studies, we have used those skin and after that we have washed with the
Okay, you have
PBS.
done that. Yeah, understood.
Yes,
Okay. And one more thing, see,
yes,
all
ma'am.
transdermal dosage forms, even if the drug is not as potent as docetaxel, there is always an attempt to apply it as a patch where you have a restricted or a definite area of application.
Yes, ma'am.
Now, this being a gel is open to spreading over any area.
Yes, ma'am.
So what is your, and accordingly the permeation, the extent of permeation, everything would change. So supposing an applicant decides, that if you're told that the dose is 1 gram, you could apply it over any area, right? When it is a gel.
Yes, ma'am.
So what is your thought process about how do you control that part of the because that would alter the extent of permeation?
Ma'am, for this delivery system that I have proposed, majorly it is more effective if we deliver it to the breast region because it will be showing localized delivery. And like you are said, the gel can be scrap off while doing something. So that was the thing I also thought, but I couldn't find.
You don't have a solution for scrapping off of the product after application. No, that is one thing. Another thing that I was trying to ask you is area. So you apply it everywhere, or you're going to say that 1 gram you apply over so much area?
Ma'am, in ex vivo studies and release studies, ex vivo permeation studies, we have applied in area of 0.64 centimeter square of the pig skin according to the French diffusion cell, the exposing area to the receptor environment compartment. So
and how much of gel did you apply?
It was loaded in carbopol 1.5% gel.
No, the quantity of gel, the quantity of docetaxel which you applied to that.
The quantity was equivalent to 200 microgram DTX. The formulation of the total was equivalent to 200 microgram DTX.
So based on that, you can make some proposal as to how much of drug should be applied to how much of area.
Yes, ma'am. According to that equivalency, we can do that.
Okay, and do you have any clue about the transdermal dose? Are there reports or anything?
No, ma'am. For docetaxel, there has been no topical and transdermal formulation. Right? Currently, I have seen the NLC also, but it was not well established. So I can say that this was the new concept. So I couldn't find the transdermal dose.
All right. Yeah, that's it from me. Thank you.
Ma'am.
Yes, candidate 5.
Yes,
it's like you have used the term ultra deformable. Why not only deformable? Why ultra deformable?
Ma'am, ultra deformable means it can squeeze 1/10 to the pore size of the skin barrier. And deformable.
You have used the term deformability index in your presentation. How did you carry it out?
Ma'am, in deformability index, first we check the size of the prepared formulation before extrusion.
Size of
the size of the formulation before extrusion, and after that, after extrusion, we check the ratio. We got the size. So after extrusion and before extrusion, we check the ratio we got.
Ratio of what? Sorry, voice was.
Ma'am,
you usually check the size of the whatever nano formulation you have made, and after that, what you did?
Ma'am, we have to pass through the polycarbonate membrane of 50 nanometer size, and after that, the size we got, which we will be doing the ratio of it before and after. Then we will be getting the deformability index.
Okay, so what is there some kind of cutoff limit that okay, this is permissible?
No, ma'am. The more we get the deformability index, indicate that the high deformability of the vesicle is there.
High reformability.
Deformability. It can deform easily to the skin barriers.
Okay, but then that also indicates indirectly that the structure has collapsed.
No, ma'am. The TAM images reveal that it has a spherical structure.
No, but then that temp image is not of. Have you actually kind of taken up that particular system which has been passed through the polycarbonate membrane for the sake of TEM analysis?
Yes, ma'am.
I doubt that.
No, ma'am. Also the ultra deformable vesicle for transferosome, transcytosome, and ethosome are well reported for its characterization. And even after the
I'm asking
deformability.
about your study, dear. I'm asking about your study. You said that you performed deformability using a polycarbonate membrane. When you carried out TEM analysis, did you use the deformed structure also and the original structure also? You have used which one?
Ma'am, it was the original one also.
That's what I'm
it
saying.
was original. Yes,
Take
ma'am.
original only. You are not going to get deformed structure. So the question here comes of Dr. Nair ki that structure was when you are dispersing this transferosome structure inside the gel, whether the drug is leaching out into the gel or not, you cannot say that with assurity, and your stability is not going to reveal that whether the structure is intact or not, because what you have analyzed in gel, I don't think you have taken carbopol gel for the sake of carrying out size estimation in the stability analysis. Have you taken carbopol gel?
Yes, ma'am, it was carbopol gel.
No, but you have prepared the formulation, but when you're carrying out size analysis in stability, how you have carried out confirm that the structure is not deformed? You have taken that gel, diluted it and perform again size and PDI.
No, ma'am.
Then how did you confirm? Madam has asked whether the structure remains intact during the process of stability or not. You have said that, okay, we have performed stability studies. But you know, even if the structure is deformed, there is some kind of leaching of drug in the carbopol gel, you cannot confirm that with the help of, should I say, size and PDI. Because तुमने तो वो जेल का नहीं किया होगा ना, जेल का किया होगा rather. जेल का किया है, किसका किया है?
For gel, we have did the characterization for pH, rheology, and spreadability to check whether the.
That is not going to give you any idea about whether the structure has collapsed inside the gel or not.
Yes, ma'am.
है ना?
I will look into it, ma'am. I will surely think about it and do the further.
हाँ, एक बार ना जब हम ऐसा claim करें that it is better that we kind of satisfy our own doubts, ठीक है ना? Exam सामने वाले के लिए अपने लिए you try to kind of satisfy those doubts of yours, ठीक है? चलेगा. That's all from my side. एक चीज और थी. How did you perform this entire rheological characterization? Which instrument you have used?
Ma'am, Brookfield rheometer.
Brookfield rheometer. Okay, Brookfield rheometer. कौन सा वाला आपने ये use किया था? Probe use किया था उसके लिए?
Yes, ma'am.
कौन सा probe use किया था कहाँ में?
I don't remember. Ma'am, actually, I don't remember the probe. I used the Brookfield viscometer.
आपने खुद किए?
Yes, ma'am. I did. Yes, ma'am. I did it, but I don't exactly remember the probe because.
एक ही की study उसमें एक ही formulation और उसमें उसी का हमने बहुत ज़्यादा rheological characterization किया है. We should remember this, है ना?
Yes, ma'am. Yes.
Another thing is you would have required large volume, right? If you used a Brookfield.
पक्का Brookfield use किया है कि?
Yes, ma'am. It was Brookfield rheometer.
Rheometer के viscometer?
No, ma'am. Rheometer. Brookfield rheometer.
फर्क पता है दोनों का visco और rheometer का? चलो कोई नहीं तबस्सुम मैडम आप पूछ ले कोई नहीं.
Okay, the very purpose of going in for your transdermal delivery was to reduce toxicity, right? So did you do toxicity assessment of your developed product or gel?
Yes, ma'am. The toxicity assessment was also done to check the different rays in the biomarkers like AST, ALT.
So I just want to know now. I just found a paper published in 2020, which is exactly what you are presenting today. So it is titled Exploring the Promising Potential of High Permeation Vesicle Mediated Localized Transdermal Delivery of Docetaxel in Breast Cancer to Overcome Limitations of Systemic Chemotherapy. This is looking exactly like what you have presented.
Ma'am, actually, this work is the allied work with my senior. So I have, yes.
Okay, fine. Thank you.
Thank you, ma'am.
Vaishnavi, our evaluation sheet mentions this candidate as OPC 006. I hope there is some kind of typo error. ये 5 ही है ना OPC?
हाँ, इसको 5 कर दो, 5 कर दो इसको.
ठीक है, चलो.
There is also OPG 6. I would like to call OPG 6 for the presentation.
So 7 to 6 करना है अब.
Okay.
Okay. OPG 6, please share your presentation. OPG 6, you're not audible.
Am I audible?
Yeah. Go ahead.
So good afternoon, judges. I'll be delivering my oral presentation on the topic Pro-Drug Approach-Driven pH-Sensitive Nanocarrier Mediated Delivery of Amphotericin B for enhancing its oral bioavailability and stability. So the rationale for choosing amphotericin B was its broad spectrum antifungal as well as anti-leishmaniasis activity, as it shows selective binding towards ergosterol, which is a component of fungal cell membrane, further creating pores and causing leakage of ions like magnesium and potassium that ultimately causes cell death. However, the drug has certain limitations such as lower solubility and permeability. It also possesses aggregation-induced toxicity. Due to such reasons, no oral delivery formulation is available till date. So we thought of an alternative to conjugate the drug with a lipid moiety to increase its intestinal permeability and decrease aggregation, which could be further loaded into liposomes and coated with gastric protectant polymer to enable efficient oral delivery of the drug. So the objectives of the presentation is to synthesize and characterize amphotericin B tosylated conjugate, then to optimize the formula for preparing liposomes, then to coat the liposomes with pH-sensitive polymer. Here we will be using Udrigit S100, then to carry out different in vitro, ex vivo and in vivo studies with the prepared liposomal formulation. So the reaction scheme, in my reaction scheme, amino group of the amphotericin B was conjugated with carboxylic group of tocopherol succinate to form a lipid drug conjugate to show enhanced efficacy and safety of the drug. The conjugate was further loaded into blank liposomes, which were prepared by film hydration method and then coated with Udrigit polymer to enable its protection from harsh gastric environment. Formulation retains its coating in stomach and coating dissolves slowly in intestine to release the liposome. The formulation was characterized by FTIR and proton NMR spectra. FTIR spectra reveals a peak at 3,408 and 1,645 centimeter inverse due to NH and C double bond O stretch, confirming the formation of conjugate having amide bond. Also, NMR spectra gave a peak around 8, confirming amide structure prevalence. Further, size and polydispersity index of the vesicles were studied. Vesicle size of the conjugate loaded liposomes was lesser than the drug loaded liposome. Also, size increases after coating the formulation.
Drug loading and entrapment efficiency was found to be higher in case of amphotericin B tosylated TOS conjugate liposome as compared to amphotericin B liposomes. Then surface characterization was also performed by TEM. So here there are three figures. Figure A shows uncoated liposomes. Figure B shows efficient coating of liposomes. And figure C confirms the intact nature of liposomes even after removal of coating. Also, size and PDI hence obtained were in accordance with the data shown previously.
Your SEM figure is not moving. My slides are not moving. I'm still seeing the reaction scheme. Is it moving for others?
Yeah, it is moving.
Okay, in mirror nets.
I think there must be a lag.
Shall I proceed with slide number 9?
No, just hold on for a minute.
Okay.
Manju, you may want to re-log in or something. Just try that.
No issues, madam. Please continue. Go ahead.
Okay.
Shall I proceed?
Continue.
With slide?
Yeah.
Okay, thank you, ma'am. As amphotericin B shows aggregation-induced toxicity, properties of the conjugate relative to the drug were studied using UV visible spectroscopy. Peak intensity of the two groups at varying concentrations were compared, and intensity was found to be lower in case of conjugates, which can also be stated as less aggregation-induced toxicity. However, at higher concentrations of the conjugate, that is 100 microgram per ml, toxicity was found, but still it is less compared to the free drug. Percent hemolysis was also carried out on intact RBCs, and coated liposomes were found to be least toxic as compared to all other groups, showing decreased systemic toxicity. Further, in vitro studies revealed enhanced retardation in case of coated liposomes. This was done using dialysis bag method, wherein the retardation in case of coated liposomes was less as compared to other groups. Ex vivo intestinal permeability studies were done in goat intestine, and it revealed enhanced apparent permeability of the conjugate as compared to the free drug due to increase in lipophilic character. Also, lower apparent permeability of the coated formulation could be justified by a slower dissolution of the coating polymer, leading to slower release of the liposomes and hence lower permeability, giving sustained action. Then cellular uptake studies were performed on CACO2 cells, which showed maximum fluorescence in case of conjugated liposomes, proving maximum cellular uptake. Cell viability studies reveal more than 90% cell viability, indicating the safety of the formulation as compared to other groups. In order for the drug to become effective, cleavage of the amide bond is a necessity, and hence conversion of amphotericin B from the conjugate was studied in liver homogenate. With increasing time, conjugate degrades following zero-order kinetics, and constant conversion rate of 2.59 microgram gram inverse R inverse was observed. In vitro antifungal activity was also studied in candid Vishwanathy cells, and the coated formulation showed higher toxicity as compared to other groups at 100 micromolars. Then pharmacokinetic studies were also performed on Sprague-Dawley rats, wherein we can see the values of Cmax and AUC of the coated liposomes was much greater as compared to the free amphotericin B. Also, the other pharmacokinetic parameters of the coated liposomes were significantly higher as compared to the marketed formulation Fungizo. So to conclude with my presentation, Udricid polymer coating over the liposomes enhanced its gastrointestinal stability and also enabled its oral delivery. Activity of the conjugate was more as compared to the free drug. Cell viability assays conclude that formulation gave better uptake and lower toxicity. Pharmacokinetic studies reveal higher bioavailability as compared to free drug by oral route. Also, this formulation is as efficacious as proven by in vitro antifungal assays. Successful formation of the conjugate with the drug not only reduced its toxicity but also improved its pharmaceutical properties. These are my references. Thank you.
Thank you, OPG6. The session is now open for Q&A.
I'll go?
Yeah.
Okay, thank you. Your prodrug is going to be converted to the drug after absorption?
Yes, ma'am, in the liver.
In the liver, then how do you explain the in vitro activity that you're getting? Like
So
say
basically,
in the candida or wherever, if the or are you trying to say that the prodrug is active as well?
the prodrug is basically not active. Here, the prodrug was basically used to increase to decrease the what we say aggregation induced toxicity and also increase the permeability of the drug.
Permeability. Agreed.
Yeah.
Agreed. But when you are exposing a candida cell or whatever to the prodrug as well as to the drug, at that time, there is no, since there is no liver homogenate or anything, there is no chance for the prodrug to get converted to the drug.
Okay.
So how do you explain the efficacy at that point?
Basically, when this prodrug is given to the species, it will increase the permeability inside the cells and thereby it will
that's
show
what.
its
So the
effect.
prodrug has to be active on its own, no? By if it is there was a experiment on candida cells, right?
Yes, ma'am.
So in those experiments, your prodrug is equivalent to amphotericin B or better. What was the outcome?
As compared at 100 micromolar, at 100 micromolar, the prodrug, it showed maximum toxicity as compared to the free drug.
So that is what I'm trying to
how
ask.
did you do the antifungal activity? How did you assess the antifungal activity in
basically they were carried out. Basically, this antifungal activity, these are the cell culture studies and usually carried out by our senior candidates to avoid the cross-contamination.
I could not hear. Probably there is an echo happening. Can you come again?
Could be checked by
I think there's a lag also.
Actually, this is checked by fluorescence activity.
Pardon?
Am I audible?
Yeah, just repeat. How did you just answer Madam's question? How did you do the in vitro antifungal activity?
The antifungal cells were observed. This
what
was
is an
the
antifungal
toxicity.
cell? There's a fungal cell.
Candid, fungal cell. So candid species were studied for the same.
How did you
study
them? How was
this done? Yeah, how was this done?
By MTT assay.
On your own, or someone else has done the study on your behalf? It is a serious work or what?
Actually, this is my allied project. The cellular studies are carried out by my senior candidates in order to avoid the cross-contamination in our institution facilities.
Yeah, but when you present, you're supposed to know everything about it, right?
This study is basically, we do it by MTT assay.
Yes, that is there. But do you know how, because in your study, the antifungal activity is a very important and crucial part. Do you have any idea about how do you actually arrive at the minimum concentration which you are supposed to use for the cell line studies? Because amphotericin itself is cytotoxic. It will cause cell death. Then you're using tocopherol. So we have to first of all titrate the dose which we could give to animals, which gives minimum 50% viability, and then we carry out our formulations further dilution to evaluate its efficacy. Do you have any idea about how you have performed this study?
Ma'am, I did not get your question.
When you carry out antifungal activity, initially you have to assess that your formulation as such is not toxic. You have to dilute it till the level the cell do not die. And then you carry out comparative evaluation of different versions of formulation which you have prepared. Okay,
Yes,
because
ma'am.
amphotericin as it is going to cause a lot of cell death. That's why I'm asking this. Do you
Yes,
have any
ma'am.
idea about that?
So like lower concentrations of the formulation was used. That is, we assessed in three concentrations, 10,
That's what I'm
20,
saying.
100.
When you say lower, you have to dilute it till the level it doesn't cause a cell death, or at least minimum 50% viability of cell is there. How did you evaluate that? Do you have any idea about how did you carry out this antifungal study? Because your entire work is based, I mean, the crux of your work is based on that only.
Ma'am, I'll look into it.
Okay, no issues.
Just one more digging here. Listen, you have done release from the liposome for the drug as well as for the prodrug. What was the method of analysis?
Release of the drug. So basically, in the drug release study was done using dialysis bag method.
No, method of analysis. How did you analyze the aliquots for
HPLC
the drug and for the prodrug? So you had to, yeah, tell me.
HPLC analysis method was used for the same.
So you have developed two methods?
Yes, for like bioanalytical method development, internal standard which I used was 1-amino 4-nitronaphthalene.
No, where did you get the reference standard for your conjugate?
Reference standard basically for bioanalytical method development. First of all, I got 3 different retention times for amphotericin B, then for the conjugate, and as well as for the internal standard. That is how I estimated.
No, but did you use a reference standard for your drug?
Yes, I used the reference standard for my drug. That is one
so
of
where did you get the reference standard of the conjugate of the prodrug?
Internal standard is different. Madam is asking for reference standard. To quantitate, you have to have the pure material against which you compare. Reference standard, not internal standard.
Okay.
Why did you need an internal standard? Let's forget the reference standard question. Why did you need an internal standard?
Internal standard, basically we need it for to check the purity of, to check if there are no other impurities in the sample. And basically
did you have
by
impurity standards also?
No, basically internal standard, it has same morphology and many properties similar to that of
structure ko. Structure doesn't have morphology, no. Cell has morphology.
Sorry, I meant to say the structural similarities as well as in certain other properties, it resembles to that of the drug. So we use it for comparison purpose. But
so will it not elute at the retention time of your drug if it is structurally similar?
No, ma'am.
Huh? Okay, can you go to your synthesis? Dr. Hema, you proceed.
No, it's fine. It's fine.
Can you go to your synthesis slide? The second slide that you did. Okay, so you formed an amide bond. What is the percentage purity of your conjugate?
I could not recall the percentage purity of the conjugate, but I confirmed it via proton NMR and FTIR spectra.
That doesn't give you percentage purity, right?
No, I just confirmed the prevalence of the amide bond in my conjugate.
So what was the yield of your conjugate? What percent yield did you get?
I'm not sure about it right now. Like I couldn't read.
Okay, fine. Thank you.
Thank you.
Over to you.
Thank you, OPG6. That was the last presentation for PG category oral presentations. I would like to quote Stephen Hawking, sir, as he rightly said, however difficult a problem may seem, there is always something you can do to succeed at it. And that's what research teaches us. When we put our mind to it, we can always succeed. I now request our judges to provide a brief message regarding the session or a feedback to our participants.
Yes, Dr. Hema, over to you.
Same sequence. Okay, so I think the numbers were few, but no, it's very good work, and it's more than that. It's a very encouraging kind of platform that you have here, wherein students can learn multiple things at the same time: how to present well, how to defend what they did well. Only one glitch, since we were in the MPharm segment right now, I realized that it's the competition is little unfair, because some candidates, the amount of elaborate work that they present is definitely, we know as teachers, it's not possible that it is purely MPharm. So that's it. It makes the competition, it may seem overwhelming and like a lot of work, but if it's not totally their own, it does make the stage a little skewed for us also as evaluators. So I think that it is difficult. I know it is difficult, but that is something that probably in the long term needs to be addressed. But nevertheless, that doesn't take away anything from the quality of the organization and the wonderful initiative of having this kind of an online student conference, because it opens the field for students across the country to participate. Geographically going from one place to the other and all may not always be possible for everyone. So this completely removes that barrier. So I think it's a wonderful stage. And it adds other barriers like these kind, which we need to be imaginative and see how we can work around it. But otherwise, it's kudos to the organizers and kudos to the presenters as well. They did a very good job of defending and presenting their MPharm work. And I've been watching the PhD presentations as well. Most of them I've sat through, so I think it's a wonderful platform. And I hope more kids, more students jump into the basket in the coming years, and more presentations we see.
Dr. Hema Nair, thank you. This is Munira.
Yeah, hi, hello.
How are you?
Good.
Yeah, thank you for your comments and feedback. You rightly mentioned that it would be a kind of
unfair.
Unfair when we have, but then it would be difficult for us to identify which one is their seniors. It would be difficult.
I totally agree.
Yeah, but you rightly pointed out this MPharm students, so what we were thinking too, so we also noted that and we discussed that next Student Research Council, we should be changing the time, because see, this September is the new batch has just started working. The earlier batch already left in June, July, so they are not going to present because they are with industry. So it is always the junior, majority of the time it is a junior who would be presenting. So we are thinking, just to solve this issue, what you rightly pointed out, we are thinking that we would be shifting the month in which the SRC we should be conducting. So somewhere where the entire MPharm thesis work is over, which is around May, month of May, we know that majority of the, at least not only Mumbai University, across, we have a student completing their thesis work somewhere in May, because majority of the industry, they would go for hiring in the month of June. So having said that, I just wanted to quickly give the stats that this time, this third especially, so till last SRC, we had 11 or 9 states participating. This time we have 18 states across India participating, and 49 cities have participated and 60 colleges have participated.
Very nice.
And these abstracts have come from 48 colleges and 35 cities and 13 states. So there is quite a diversity in terms of institutes also. And we could see today in PhD and MPharm work, we could see good quality data and good work in lot of papers which students presented. So I think we are good.
Yeah, there's one more thing. Your idea of changing the timing is great. I think that is definitely an issue, that the person who has done the work has already passed out, is working and is not available for presenting. Also, I have a feeling looking at these that, I'm being very frank, in that very large institutes, where the groups are working together, PhD and MPharm, some of this work is not only MPharm work.
Yes.
So that's also, I mean, that really, it adds to the unfairness, but I think that's life. So we have to take it.
Yeah, and it would be very difficult to identify that.
I totally agree. I couldn't agree with you more. It's very difficult.
Yeah, thank you. Thank you very much.
My pleasure.
So
I have to thank you for having me over. My pleasure, totally.
In fact, I'm thanking all the three judges, Dr. Hema, Dr. Tabassum, and Dr. Manju Mishra. It was great having you on our online platform. Good inputs are coming. And it's quite a learning experience for all those participants who are not presenting. It's the design of experiment, the data, methodology, data analysis, the way it has been presented, the way it has been concluded. That's all entire exercise is a learning exercise. So thank you very much, Dr. Manju Mishra madam, Dr. Hema madam, and Dr. Tabassum.
Thank you. Dr. Manju, you want to comment something? I think Professor Hema and Dr. Munira Mamin have aptly summarized everything what I wanted to say. What I could suggest is that we have been also conducting this kind of event. And what I could suggest is that probably we could not be having this differentiation of PhD and MPharm. Rather, we could have themes of, say, probably from pharmaceutics or pharmacology or something like that, where we could have poster session, because I could clearly tell you from the presentation that some of the work which they have presented is not at all possible in the MPharm duration. It was a part of a big, larger group, which is normally the case with almost all bigger established research institutions where they have group. So they present each other's work. It is not even a single senior's work. It is a combination of 2-3 people working in that same group which they have compiled and presented. Because most of the time they were not even knowing what was the make of the instrument, how they have performed the study and like that. Okay, so it's obviously, as Professor Nair has pointed out, that this was unfair on part of we comparing PhD versus MPharm work. So probably we could. Dr. Munira Madam has already pointed out, it's difficult to identify which work is MPharm, which work is PhD. So that's absolutely there. We just could not identify, but nevertheless, it was good work. A lot of presentations were too good to be kind of considered to be an MPharm work. There were other PhD work, I must say. But anyhow, it was good presentation. This kind of forum is definitely good for students. They get to know how to present their work, how to learn from it. So it was good. Overall, a very good attempt for the students and by the university. Thank you for inviting me.
Thank you. Do we get together to score, or do we independently score? What is the thing?
Who's Vaishnavi?
The question? I was not. Yes, ma'am.
Vaishnavi, can you just ask people to leave the room, then we can just discuss maybe just 2 minutes, not more, because there are just 4 entries.
Because it was a little complex, so.
Okay, ma'am. Should I ask to rejoin them again?
I think there are too many people. Is it difficult?
Yes, ma'am, actually.
No, do you have something planned, or this is the last thing? If this is the last thing, people will eventually leave.
Actually, ma'am, I have a concluding message, and then eventually the people will leave. This is the
so you'll give the leaving message, then you leave us alone.
Okay. Okay. So thank you so much to all the judges for providing their such valuable, insightful messages. And now I will just a second. I would like to present judges this small token of appreciation from us. Thank you, Dr. Hema Nair, ma'am.
Thank you. Thank you so much.
Thank you, Dr. Manju Mishra, ma'am. And thank you, Dr. Tabassum Khan, ma'am. Again, I would like to thank all the judges for being such a vital part of the event and providing the participants with such valuable feedbacks, which have widened our horizons and provided us with new perspectives. This knowledge will help us to make each day a little better than before. We also thank all the participants for the active participation and the audience for their kind patience. Everyone is requested to fill the feedback form today itself. The link will be posted in the chat box. So with this, I would like to conclude the day one of the third Student Research Congress. Also adding one more point, the feedback form link will be also posted in the Telegram group. So it's requested to fill out the form. Thank you so much. Okay, ma'am. So we'll all leave.
Yes,
thank you so much, ma'am.
Good job, hosts.
Very nice.
Vaishnavi and
yeah.
Vaishnavi and fill in the blanks, Vaishnavi for me.
Sandhya, ma'am. Sandhya.
Sandhya.
Yes.
Vaishnavi and Sandhya.
Yes, ma'am. Thank you so much. So everyone can leave the meeting, the participants and the audience.
You be the last person to leave just to ensure that everyone is left, okay?
Okay. Okay, ma'am.
बंद कर दो स्ट्रीमिंग चालू रहने दो बंद कर देना यूट्यूब पे जा रहा है सब